So welcome back, everyone. Today we're going to have two topics, but starting with a keynote from Marc Chadeau from Imperial, and then we'll have two sessions. One about more machine learning tools applied to the exposome data, and the second one at 5, it's going to be about causal inference and the use of omics data to infer relationship between the exposome and health outcomes. So we're starting now with a seminar from Marc Chadeau from Imperial College, professor of computational epidemiology and biostatistics. He actually started at Imperial in 2005 and has been working on the integration of omics into the complex association between exposure and health. He was in the exposomic project and now in the EXPANSE project in the same network as a European project athlete that LIRIS-ISGlobal is leading. So the floor is yours, Marc.
Thank you very much, Leah. So I will share my screen. There you go. Can you see my screen?
Yes, we can see.
Yes, and it's going to move on full screen now. Yeah.
Good. Yeah.
So thank you very much, Leah, and for the opportunity to give a talk there. So as you said, I'm going to report on some work that we've done in the past in previous exposome projects and what we're currently doing within EXPANSE and within Longitools. So as you know, I'm not gonna spend a lot of time in defining the exposome, but there's quite a few definitions of the exposome. But one can broadly consider either the internal exposome, which is the ensemble of molecular responses to external stresses, and the external exposome, which you've seen quite a few presentations already, which relate more to the families of exposures and try to look as an entirety these sets of exposures. So the first efforts that was done in those primary exposome projects, so namely the Exposomics project and the Helix project, and we collaborated those two projects together to come up with a kind of reconciled list of models that one could use to explore the complexity of the exposome. So on the one side, we had the internal exposome, which was featuring mainly molecular data, so with a high P and low N situation, and complex correlation structures to be validated for. And on the other side, the external exposome, which was the focus of the Helix project, where the question was lower dimensionality issues, but more block correlation and complex correlation structures to be accommodated. And in both reviews that we completed, we did some testing of the different approaches that could be used. And irrespective of the way we considered the exposome as the internal or its external components, we really thought that the use of viable selection techniques was outperforming other approaches. Now, when you're talking about exposome analytics, I think it's good to see it as a mixed culture from different fields of quantitative sciences, including statistics and machine learning. And so basically the objective of exposome analytics has one big pillar, which is understanding and modeling. So more focusing on risk factors. So first of all, of course, identify features and variables that are associated with an outcome of interest and or of an exposure of interest, and try to estimate their effect and their link and their correlation with other signals. One key issue that we're facing in exposome analytics is interpretability. So we need to account for redundancies within the data. So that leads us to more sparse models and to infer mutually adjusted effects. So that's more the multivariate approaches. Then of course, in that more etiological context, what's really fundamental is to get some functional insights into the associations and signals we detect. And to do that, we need to explore the underlying mechanisms from observational data. So that's a kind of gold standard for these is to move to causal inferences. But biological interpretation and biological investigation is also helping in that matter. Then there is another second big pillar, which is about prediction, and mainly to quantify the role of these risk factors. So the underlying assumption is that those validated associated features should contribute to the explanation or the prediction of the outcome. And therefore, it leads to some estimation problem to estimate not only the marginal effect of these factors and their contribution to the prediction, but also their joint and possible redundancy. So with this slide, I'm hoping to show that there is a balance between interpretability and predictive performances that we're trying to achieve when we're doing exposome analytics. So from this mixed inheritance in the approaches, there's also a mixed inheritance in terms of data. So as we've highlighted in the first slide, you have some big data coming from molecular and population sciences, where you have either wide data with a lot of measurement in a few individuals. So that's typically what you encounter when you're doing molecular epidemiology. Or you can also have high data with a very large number of individuals included with lower resolution data. So in the first case, you are in a high dimensionality problem. And in the other one, it's more about summarizing the information and try to identify latent population structures. At this stage, and currently we're in a situation where we need to integrate these complementary and heterogeneous approaches to get a better picture and exploit as much as we can the wealth of data that is accumulating in the field of exposome research. In terms of techniques and algorithms, there's one big issue about explicitly and interpretively model our data. And so regression-based approaches are usually used in the penalized version. Also, another angle that is taken is to reduce the complexity of the data via supervised or not dimensionality reduction techniques combined with clustering and classification approaches. And of course, but this is more exploratory, there is some non-parametric alternatives that needs to be accounted for to basically move a bit away from the usual linear assumption that we're making in all these regression exercises. So what I'm going to talk about during this presentation is really to see how we can go beyond association studies. And I'm going to illustrate that in some of the exposome studies, what we really had to face and to take into account is weak effects and subtle effects that we need to boost our power, statistical power. And to do that, we implemented complex study design. So I'm just going to show an example on how to extend the established approach to accommodate this. Then also we need to accommodate complex responses. And so this is more looking at block modeling and trying to take as a prior that the data that we have belong to different groups and different blocks. Then the second angle I'm going to try and illustrate is how to get a bit further, a bit closer to getting an insight, functional insights into the discoveries that we're making. So I'm going to show you two examples on the modeling of life course exposures, taking the example from smoking, and try to integrate multipolomics data to try and get more mechanistic insights of what is the effect of these long-lasting exposure. And finally, I will conclude, depending on the time, on some feature selection exercises that we've done for risk modeling, so using group penalization. So very briefly, to start with, an example that stems from the Exposomics project, where we had to implement experimental studies where we basically control the environment and assess the variation in molecular profiles in relation within a given individual in relation to the experiment. So in that case, it's just a matter of separating out the intra and inter-individual variability and focus on the intra-individual variability, which is capturing, we're assuming, the response to the experimental changes. So to do this, the usual way of looking at that is to use mixed models, but in the case of very complex designs, that might not be ideal, and you might want to make sure that you model the variance covariance matrix of the observation in a more complex and more exhaustive way. So one approach that we've proposed for the univariate setup is to look at multivariate normal models where you have that variance covariance structure that depends on the experimental conditions. And also, we implemented some multi-level, multivariate approaches to accommodate multivariate regression models. So I'm going to show you some examples of a simpler study design, which is the Piscina study, where we had, which was carried out in Barcelona. And that was a 60 participant that swam in a chlorinated pool. And we measured in these 60 individuals their exposures to disinfection byproducts before and after the swim. And we measured in these people several omics profiles, including metabolomics profiles, in which we had, from that platform, 6,000 metabolic features assays. So this is, in a nutshell, what we found with our multivariate normal model. And we find here, we represent here a volcano plot showing the associations that we identify for each of the 6 measured exposures. So overall, we identify a bit less than 300 features that were associated with at least one of the exposure. And what was very striking from this plot is that there was a strong overlap between the exposure associated feature, with more than half of the features that were associated with more than 3 exposures. And this was not only detected at the transcriptomics level, but also at the proteomics level. And not only at the metabolomics level, but also proteins and transcriptomics level. So the conclusion was that the effect was quite a-specific of the different exposures we were measuring, which could, and this is what we're summarizing in this plot, be at least partially explained by the fact that these were strongly co-occurring exposure during the swim, actually. So the question we had there was whether that strong overlap was just due to the fact that these exposure co-occurred. And in this paper, we decided to move for a more extensive analysis of the data. So we took not the metabolomic data there, but as a proof of principle example, the proteins that were measured in these individuals. So that were 13 inflammatory proteins. And we looked at block models, so in that case, partially squared and the sparse alternatives, to try and see first if all the exposures were predictive of the 13 proteins. Second, to try and see within those exposures, which were the ones that were driving a potential proteomic signal, and within the proteins, which were the ones that were mostly explained by changing the exposure. And we accounted for the fact that we had repeated measurement by applying a multi-level extension of the PLS, which is pretty much computing the intra-individual variability and pretty much performs a PLS model on that intra-individual variance. So this is, in a nutshell, what we found in this paper. So this is the result from the PLS. This is a result from the PLS when we're inducing sparsity in the exposures, which are the predictors. And this is the results of the model where we induce sparsity in the response. So here we can see that despite the strong correlation and co-occurrence of the exposures, that the bromoform, the blue bar here, was not found to be selected as being adding anything to the 3 other exposures to predict the set of 13 inflammatory proteins. Conversely, if you look at this plot here, we're showing that the 4 proteins are not explaining all of the, the 4 exposures are not predicting all of the 13 proteins and explain mainly 4 or 5, the variability of 4 or 5 proteins. So pretty much we found in that analysis that modeling not only the correlation among the predicted, but also among the response is helping prioritizing which are the influential exposures within the mixture, and second, which are the mostly affected sets of proteins. Now I'm moving to the second questions that we had, which was more to relate with lifelong exposures. So I'm taking very quickly this example of that famous plot from Peto, which is showing that in former smokers, the risk, even years after smoking, stopping smoking, does not get to the risk of lung cancer of someone who never smoked. So the idea here is that lifelong or past exposure to smoking may have left a biological trace. And the question we had in this paper was to try and see whether we can detect some methylation signals that would support the fact that there is a residual risk due to smoking for lung cancer years after smoking cessation. So this was based on 750 individuals in which we had measured the methylation via the Illumina chip, and we ran a series of epigenome-wide association studies, and we did some recoding tricks to look at the dilution effect as a function of time since smoking cessation. And we identify from this plot, here we're showing the CPG sites that are associated at least in one of the 40-ish EWAS that we're performing. And we're listing here the ones that are associated at least once. And here we look at when they lose statistical significance in the recoding. And from this plot, it is apparent that you have two classes of markers. The first one that tends to lose significance after several years after smoking cessation, and some that remains associated even 40 plus years after smoking cessation. So we've got those two classes of biomarkers. When you deal with methylation data, it's always a bit complicated to move from the signal you identify to their function. So what we decided to do in that paper was to relate those 750 CPG sites to a genome-wide transcriptomic profile that we had in a subsample, and did a very simple one-to-one pairwise association study investigating almost 7 million pairs. And of those 7 million pairs, we identified a bit less than 6,000 that were found significant. And what was striking in this paper was that of these CPG transcript pairs that were found significantly associated, only 5 involved the CPG and the transcript that belonged to the same gene. So that clearly identified that the epigenetic regulation of gene expression do respond to complex and possibly non-local phenomena. So that was if you want, a justification for us to move into a more global view of the correlation patterns between the CPGs and the transcripts. So what we decided to look at is to take the 426 transcripts that we identified in these pairs and 265 CPG sites that we identified in these significant pairs, and try to look how they correlated into a partial correlation network. So we had a bit less than 250,000 edges to explore. And if you are to run classical calibration procedures based on, for instance, Bayesian information criteria, you end up with that big blurb of a network that is totally uninterpretable and very separate, where you have in blue the CPGs and in red the transcript, and you can see that pretty much they do not talk to each other at all, which is due to spurious correlation structures that are within the blocks of data. So we defined a bespoke multi-block calibration strategy based on stability analysis. And this is the calibrated network that we ended up with. And what was very interesting in this work was that we identified here a very central role of LRRN3, which was one of the 5 CPGs and transcripts that were associated, and that was the only one that was associated to smoking, both at the methylation and CPG level. So it reproduced quite well what we identified. And from this type of network, you can also use the fact that gene expressions are well functionally characterized, and you can link the nodes or the transcripts, if you want, to the pathway they belong to. And for instance, clearly here, if you look at the transcripts that are related to the stress response pathway, you can see that they cluster together pretty well. And therefore, that this could give you some functional insights into the CPG sites that are in the neighborhood of these modules in the network. So this is another example where we're using feature selection for disease prediction. So that's a paper that we've done using 92 inflammatory proteins measured in a similar study population. And in a subsample of these individuals, we also have transcriptomics data. And in all of them, we also have metabolomic data. So the angle we took for this was to use a multi-variable selection approach to try and see whether we could find some proteins who are mutually and complementarily associated with future risk of lung cancer. And this is what we identified with LASSO, but what we decided to complement this analysis with was to use, again, stability analysis and try to identify among those the proteins that didn't seem to be associated through potential outlying observations. And here we report the selection proportion of different proteins in the base model and in a model that was adjusted for smoking as measured by pack years. And we clearly identified that CDCP1 was strongly associated. And we identified a series of other proteins that were also jointly associated with the lung cancer outcome. And some of these associations were not detected if we were to run a univariate model. Here is just to try and see whether these molecules that we've identified were adding to smoking in terms of predicting lung cancer. And we could see that there was a moderate increase, but still an increase in the predictive performances when we added the blood level of CDCP1 in addition to smoking. When we used the lasso selected proteins, which were more numerous, we also included, but very marginally, the predictive performances. So this analysis showed that there was a complementarity of the proteins to predict lung cancer and that they were complementing smoking. So not only were they etiologically interesting, but also they contributed to the prediction. Now, in these samples, we also have measured metabolomics data. So we have the metabolic profile that was acquired in different ionization modes. That was data that was generated at IARC in Augustin Skalberg's group. And so you can see here, what I referred to is that you've got some strong correlation pattern within platforms. So these are the positive mode, negative mode, and this is the bipartite correlation patterns. And you can see that you've got within platforms stronger correlation patterns, which is why we need to have that block correlation, that block calibration implemented. So in order to improve computational feasibility and to improve interpretability, we opted for the following approach, is that we took our metabolomics profiles, and we first ran a hierarchical clustering on these to identify groups of metabolic features that were highly correlated. And within that group, we decided to summarize the cluster by PCA and use the first component of the cluster. So that clustering exercise was calibrated to ensure that the first PC was explaining at least 70% of the variance of the cluster. So we ended up with a bit less than 1,400 clusters for the positive mode and 700 clusters for the negative mode. And basically, we run the same stability selection approach for the correlation. And here we're looking at those in blue for negative and in red for positive mode. And you can see that there is a sparse representation and sparse interconnection of the different models, of the different modules. And you can see here that P361 is a module that seems to be quite central and related to many others. So if we were to prioritize a set of metabolites, we could focus on those. Then we extended this and also looked at the lung cancer related proteins that we identified before and CpG sites that were reported in previous publications, and ended up with that multi-block correlation networks that again deserve some further and in-depth investigation, but the topology of the network and the centrality of the different nodes and their relatedness is quite indicative of the possible functional insights of those metabolic clusters and the CpG site they correlate with. So this is for the omics integration. And now I'm just going to give you a very brief example of what you could do in terms of complexity reduction. So the underlying assumption is that you could use some low resolution markers or biomarkers and include those as markers of aging. So here, this is what we've done in the UK Biobank. So I'm sure you're all very familiar with the UK Biobank, but in a nutshell, it's a huge study of 500,000 individuals with in-depth characterization of the individuals and very up-to-date mortality and incidence data, and in whom we have genome-wide scans that enables to do some GWAS and also some MR, but I'm not going to touch upon this. But also what's interesting for us here is that we have in these participants a set of 30 prioritized biomarkers that are enabling us to measure biological aging using established goals. So in terms of health outcomes, you've got many outcomes, but if you look broadly, you've got quite a few mortality outcomes in cancer, CVD, external causes that you can refer to, and you've got a very large number of incident cases for CVD and cancer. So in this paper, we decided to use these biomarkers to try and see whether we could use composite scores such as the biological health score, or which is an extension of the allostatic load, which pretty much is just if you want, a measure of the physiological wear and tear of different physiological systems that includes cardiovascular system, liver system, and metabolic system, and so on. And so basically the idea behind this is that your biomarkers level are representative of what you've been facing throughout your life. And if you have some of the systems that have been solicited too much due to external stresses, that would be captured by your levels of these biomarkers. And these composite scores are very simple to calculate and are meant to capture that physiological wear and tear. So that's the first question. Can we compute composite scores, and are they socially or are they unevenly distributed across the population? First question. Second question, are they related to health? So to do this, we ran some survival models in the UK Biobank, and we're just going to show some results on incidence. And the third question, which is more exposome related, is can we identify some molecular support for potential associations? So in this slide, I'm just showing the distribution of the score, so that health score, and you can clearly see that there is an unambiguous gradient of the score in relation to education. So you've got a higher score for the low education individuals and a lower score, i.e. a lower risk, in the highest education group. And you can see that this gradient is unambiguous in men and in women and across all age groups. So this you can do in many studies. We have another paper that looked at it in another study, and we've replicated these findings in many studies. But what we could do in UK Biobank was to relate those and just to simply explore the distribution of that score in the full population in black, and in blue here, you have the distribution in people who went on to develop a CVD event. And you can clearly see that there is a right shift in the distribution of the score, which is indicative of a potential association. So we investigated that further and formally using a series of Cox models, where basically we looked at the score and then subsequently adopting a path analysis strategy on time-resolved exposures. So basically we adjusted on education and on behaviors, BMI, and on medical history. And we represent here the results for the BHS in pinky, then in blue the metabolic score, cardiovascular score, and so on and so forth. And what's clear from this is that you have an effect of the score on cancer and CVD incidence, but most of it is explained when you adjust on indication and behavior for cancer. But when you look at CVD, there is a drop and there is an attenuation when you adjust on BMI, but it remains statistically significant and associated with the outcome, even in a fully adjusted model. So there seems to be an additional element that is captured by the score that could explain the incidence of CVD. And if you look at the subscores, it's almost the same results for all the scores except for kidney. That doesn't seem to be too associated. So here is a very quick plot that is looking at the prediction performances as measured by the C-stats. So that's what you find in the bottom table. And you can see that the BHS is as predictive as behaviors when it comes to CVD incidence, and that it complements the education and the behaviors which are established drivers of CVD incidence. So the question we have is that there seems to be an element that is captured by the BHS that is not related to education, despite the education gradient that we observe, and by behavior. So can we find a biological support for this complementarity, and can we explain it? So we basically transferred this score to the North Finnish birth cohort, which is a Finnish cohort of including 12,000 mother and child pairs, where we have individual characteristics and biomarkers measured at baseline and repeated. And from these data, we were able to capture the 5 physiological systems that we assayed. And we also had in these people a set of 235 targeted and annotated NMR data that were defined in 2 groups of molecules. And we also had DNA methylation and protein measured in these people. So we basically first computing the score in these individuals, and second, try to see if we can find within the metabolites some signatures of the score. So this is what we find here. And we've run for this a sparse group PLS model where the outcome was the BHS, so the total score. And here you've got the different families of metabolites that were assayed. And we look at the ones that are selected to predict jointly the score. And you can see that we've selected 43 different metabolites that belong to 7 out of the 10 groups that was assayed.
Marc, if you can wrap up.
Yes, I will. So again, you can look at sub-scores and have some further insights into what they relate to in terms of molecular signals. So I'm going to drop this, but this is just to show you that this is not only related to those low resolution scores. And here's just an explanation, an illustration of how you can extend this using polygenic risk scores. And this is the follow-up paper of a paper that we've submitted last year to, we published last year in JAMA. And basically here we're showing that some of these routine biomarkers are also contributing and adding to the prediction of CVD incidence. So basically, the summary of these analyses are showing that those composite score and low resolution score are capturing some biological and social gradients in the population, that these can explain the incidence of several conditions and also mortality, and that they complement established risk factor. We've seen that they are exhibiting some interesting and group correlation with molecular features. So that is a way into the potential pathways that may be involved. And now when it comes to apply this to exposome research, basically the idea is to move from scores that are defined a priori using an a priori subset of biomarkers, is to use exposome features as clustering variables and try to identify groups of exposures that tend to stratify the population and increase the contrast of their exposome experiences, if you want. And then once you have this, the idea is, in this we call them the expo types. The idea is to identify first, what are the main exposures that are driving these expo types? And second, what are their biological correlates? And finally, an ensuing question for all of this is to try and disentangle possible causal and mediating role of the biomarkers in the exposome to health association. So all of these ongoing research is part of the Expanse and Longitudes project. And as a wrap up, there's always a discrepancy or two schools that are fighting in statistics versus machine learning. So which I prefer to cast into an etiological versus prediction opposition. But I guess, and I hope that this presentation is highlighting this, that when we're doing exposome research, not only are we interested in etiology and prediction, but what we're trying to get is really to get the most of both the statistical, which is more focused on etiology and causal assessment, and prediction on the other side. And I think that the exposome analytics is really a nice merge of those two approaches. I think I'm done. I'm sorry if I rushed it. I realized I had quite a few slides. Just wanted to acknowledge the people working actively on all this work from our Imperial team, but we also collaborate with the INSERM on this and with colleagues in Utrecht. And I also wanted to acknowledge the support for the Past Life Path project and the Expanse and Longitudes project, which are ongoing H2020 projects. I thank you for your attention, and I'm sorry if I'm in time a bit.
Yes, thank you very much, Marc. I think you very well illustrated how exposome data is not just, you haven't just done an exercise of data crunching, but really answered important research question on mechanism, but also how they can be used in for better clinical like lung cancer prediction. And also I really like the work you showed on the composite score. I think that's a very nice application. We don't really have time for questions, but I have one burning question. How do you see this composite score being applied in different cohorts? Because you will always have missing variables moving from one cohort to another. How do you still implement these scores in different populations?
That's a very good question. And the thing is, and as a statistician, it was a bit of a sad realization, is that these scores are really brutal. I mean, it's dichotomizing continuous variables all the way. And first of all, we were able to reproduce these gradients in many, many studies, irrespective of the number of biomarkers we included in the score, and irrespective of the way each of the systems were measured. Because it could be the case that in one study you have, say for inflammation, you've got CRP, and some other you have another molecule. And it seems that because it's low resolution and because it is meant to be quite rough estimate of the physiological system, it reproduces quite well. And I was stunned to see, we tried many alternatives, like more quantitative alternative. We did, of course, some PCAs, we did some more trees and some more machine learning algorithms to try and get the best of the biomarkers. And none of them outperformed that very knowledge-based score, actually. So I think the fact that it's not really specific to the biomarkers it includes makes it more transportable than other scores.
Okay. Well, thank you very much. Maybe you can take some questions from the chat. If people have questions to you, you can answer by writing. And also we have the special chat, the networking break after the first session. And there will be a room where you can join, and if Marc, you can join also and answer question in this space. So that will be later on. Also, you can see in the chat, Juan Ramon commented on sharing the codes. We already have a lot of participants that have put their code in the GitHub repository. If you have any problems, please let us know. Yeah, you can contact Juan for a question regarding this. And I leave the floor now to Paola. And just a reminder also to vote on the presentations. Rodney will put the link in the chat. Thank you, Marc, again for your great presentation.
Thanks for the invitation.
Hello, everyone. Hi. My name is Paula Petrone, and I work at LIRIS-ISGlobal. I lead the biomedical data science team. And first, I want to thank the organizers for this fantastic hackathon and all the speakers and people that have prepared this great work. So I'm now going to introduce one of my favorite sessions, and I'm, of course, biased because it's machine learning and data science, which is my field. And so to start, we're going to start with Sejal Mistry from the University of Utah. He or she, Sejal, is going to present clustering exposure trajectories to classify phenotypic characteristics. So the floor is yours.
Thank you for that introduction. Just give me 1 second to share my screen. Okay, and can everyone see that okay? All right, then I will get, oh, it's looking okay?
Yeah, we're okay.
Okay, great, thank you so much. So I'll go ahead and get started then. So good morning or good afternoon, whatever time zone you're in. My name is Sejal Mistry, and I'm an MD PhD student at the University of Utah in the Department of Biomedical Informatics. Today, I'll be presenting work on how our group uses machine learning clustering methods to classify phenotypic characteristics. Specifically, I'll be sharing work on how we identified prenatal and postnatal exposure clusters and their role in pediatric obesity. And so I'm going to start off with a quick introduction to obesity as well as environmental exposures. So obesity is defined as the abnormal accumulation of fat cells or adipocytes in the body. The accepted clinical measurement of obesity or overweight is the body mass index. An estimated 21.2% of adolescents, 20.3% of children, and 13.4% of infants are classified as overweight or obese. Overall, raised BMI is a major risk factor for several non-communicable diseases, which is why understanding why obesity occurs and the triggers for it is incredibly important. So environmental exposures have recently been implicated in the pathogenesis and development of obesity. Obesogens are exposures that disrupt normal metabolic processes and increase susceptibility to weight gain across the lifespan. Several common exposures, including organic pollutants, tobacco smoke, and prenatal antibiotics, have been associated with obesity in animal as well as human models. Notably, however, these studies focus on the role of single exposures, and only a few studies explore the combination of multiple exposures on obesity risk, and how multiple exposures can act as obesogens still remains poorly understood. Additionally, only a handful of studies evaluate the role of both prenatal as well as postnatal obesogens in the development of obesity. And so taken together, there's a clear need for understanding obesogens across the lifespan and how multiple exposures interact to increase risk for obesity. And so to better understand the role of multiple obesogens prenatally as well as postnatally, we aim to determine the role of transitions from prenatal to postnatal exposure profiles in pediatric obesity risk. Our three sub-objectives were to cluster prenatal exposures using machine learning methods, to characterize the linkage between prenatal and postnatal exposures, and then finally to identify statistical associations between the identified cluster transition groups and obesity risk. And so now I'll outline our methods. So this is a summary of the methods that I'll discuss in this talk. I'll start off by talking about the pre-processing, and then I'll present each of those kind of sub-objectives with their methods as well as with their results. So the first step of our methodology was pre-processing. We mapped prenatal exposures to postnatal exposures. And so we did this by taking all of our exposures and seeing where the similarities were between the prenatal and postnatal ones. We identified 56 general exposures that span lifestyle and chemical exposures. 13 exposures were specified at a location, and so we chose the maternal as well as postnatal home exposures, and there were 13 of those. And then finally, there were 6 exposures that were specified at a time, and so we chose the 1-week time point for this analysis. And so a total of 75 exposures were included. We then label and coded categorical features and applied a min-max scaler to standardize the data. And so now I'll go through each of the research objectives. So the first objective was to cluster prenatal exposures. We first performed dimensionality reduction using principal component analysis. Next, the principal components themselves were clustered using k-means unsupervised clustering, and this is for the prenatal exposure specifically. And then finally, the optimal combination of principal components and clusters was identified using the ELBO method with the internal evaluation metrics of the silhouette score, which measures cluster compactness, as well as the Kalinske-Harabaugh index, which measures cluster separation. And so when we performed the dimensionality reduction on prenatal exposures, we plotted the cumulative explained variance, and we found that 80% of the variance was explained by 18 principal components. So we then tested principal components ranging from 1 to 18 and clusters ranging from 2 to 20 using this ELBO method. And so the ELBO method identified 3 principal components with k is equal to 4 clusters as the optimal combination. And you can see this in the figure on the left here with the Silhouette score having a peak here at k is equal to 4, and then here in the Kalinske-Harabaugh score having a peak at k is equal to 4 as well. So when we plotted the principal components across the eigenvalues for each feature, we observed, as you can see in this diagram here, that each principal component is being driven by a unique subset of exposure variables. So overall, these findings suggest that k-means with 3 principal components and 4 clusters is an appropriate model for clustering prenatal exposures. The next set of methods surround characterizing the linkage between prenatal and postnatal exposures. We use the machine learning model developed on the prenatal clusters to predict cluster membership of the postnatal exposures. Next, we characterize the distribution of each of the principal components across each cluster. And then finally, we conducted a post-hoc analysis to identify significantly different principal components using the non-parametric Mann-Whitney U statistic with Bonferroni correction. And then we also looked at effect size using Cohen's D statistic. And so we'll start off by discussing the prenatal clustering and how that performance looked. And so when we evaluated the clustering of prenatal exposures, as we can see in this figure on the top, each of the clusters separate well in space along the three principal components. In the lower figure here, we generated a box plot to highlight the distribution of each principal component across each cluster. So the star here indicates a significant difference at the 0.05 significance level with a large effect size defined as a Cohen's D greater than 1.5. So as you can see here, principal component 1 is significantly elevated in cluster 1, principal component 2 is significantly decreased in cluster 2, principal component 2 is significantly increased in cluster 3, and then finally component 3 is significantly increased in cluster 4. And so together these findings tell us that each prenatal cluster is being driven by a unique principal component. We did the same thing for the postnatal exposures that were predicted using this prenatal model. And so as you can make out from this diagram, the clusters also separate well across the three principal components, but you can see here that there's some overlap between clusters 2 and 3 that wasn't necessarily present in the prenatal exposure clustering. So similar to the prenatal finding, postnatal cluster 1 is driven by principal component 1, while postnatal cluster 4 is driven by principal component 3. However, there are no principal components that were statistically significant in clusters 2 and 3. And so taken together, these findings indicate that principal components adequately and similarly separate prenatal and postnatal exposure clusters. So our last objective surrounds determining the role of transitions from the prenatal cluster membership to the postnatal cluster membership. So to do this, we first identified prenatal postnatal cluster transitions. We then tested the association between cluster transition group membership and obesity risk, and this was done using a binary logistic regression model, and we also calculated the odds ratio for each transition group. Finally, we identified key exposures that were associated with cluster transitions that represented increased or decreased obesity risk, and that was done using the Mann-Whitney U test with Bonferroni correction for continuous exposures and the chi-square test for independence for categorical exposures. And so the first thing we did is we visualized how individuals transition from their prenatal cluster assignment to their postnatal cluster assignment. And so as you can see in this Sankey diagram over here, on the left, we have the prenatal cluster assignment, and on the right, we have the postnatal cluster assignment. And so we can see that several individuals remained in the same cluster prenatally and postnatally, but we do see all of this kind of variation here that indicates that people are indeed switching their cluster membership. Prenatally compared to postnatally. Our binary logistic regression test was performed to identify that association between each of these transition group memberships and obesity risk. So here we defined obesity risk as having an obesity category of overweight or obese. The transition groups that were significantly different are presented here. And so as you can see, the transition groups 3 prenatally and 3 postnatally, 3 to 4 and 3 to 2, here in the red, are all significantly reducing the odds of obesity with these lower odds ratios. And then in comparison, we can see that clusters, transition groups 2 to 1 and 1 to 1 significantly increase the obesity risk, and that's these blue highlighted cells here. And so taken together, these findings suggest that prenatally being assigned to cluster 3 is protective of obesity, while postnatally being assigned to cluster 1 increases obesity risk. So the last thing that we did was we tested the association between individual exposures in this quote unquote harmful group compared to this helpful group. So from our original principal component analysis, we selected the exposures that had an individual eigenvalue of 0.25 or greater, and that resulted in 11 exposures for comparison. So first I'll present the results of the continuous variables. And so here an asterisk indicates a large effect size as well as statistical significance at the 0.05 level. And so NDVI, which is the normalized difference vegetation index, was significantly elevated in the helpful group compared to the harmful group, both prenatally as well as postnatally. And then temperature was significantly decreased in the helpful group compared to the harmful group, but that was only prenatally. For categorical values, which are presented here, we report results with a large effect size defined as a Kramer's V greater than 0.5. And so here, as you can see, prenatal cereal consumption, prenatal access to public transportation, specifically the bus lines, the bus stops, as well as postnatal public transportation access with the bus stops was significantly different among the helpful and harmful groups.
We need to wrap up, Sejal.
Okay, so overall, we identified prenatal postnatal cluster transition groups that were associated with obesity risk and identified exposures that were differently expressed between these groups. And so just to quickly hit on the discussions, our main findings were that we used a k-means unsupervised machine learning model with 3 principal components and 4 clusters to characterize prenatal and postnatal exposures. We identified subsets of prenatal postnatal cluster transitions that were associated with increased as well as decreased obesity risk. And then we actually identified certain exposures that correlated with those groups. And then overall, there are several limitations to this work. We did not integrate genomic data, and we also used a subset of variables. And so including those things would be beneficial for future works. We also hope to adjust our findings for covariates and further evaluate principal components across each cluster. And then finally, we also hope to integrate metabolomic data. And so here are a list of references, and I want to thank the organizers of this conference as well as our lab group, acknowledged here as our various funding sources. And I want to also thank the Sejal lab group for their guidance in this work. And I'm happy to take questions.
Thank you so much, Sejal, that's great. Questions from the audience? Well, I have one question. If you have to think about these clusters, what do they have a meaning by themselves? What were the exposures in each cluster? For example, were all they related to transportation, or do they have some patterns in these clusters, or were just random exposures?
So the exposures that we chose to use for this analysis were kind of spread across lifestyle variables, transportation, meteorologic variables and things like that. And so that is kind of one of our next steps, to go back and look and really well define each of these clusters. And so here, for the sake of kind of time, we kind of presented the results related to the specific exposures. And so those temperature, the NDVI, so the vegetation index, and the access to kind of public transportation in the bus routes were what popped out initially.
Thank you. Anyone else? One more question? All right. Thank you so much, Sejal.
Thank you.
We are going to welcome the next speaker, John Pearce from the Medical University of South Carolina. He's going to talk about exposure continuum mapping for predicting health and disease in exposome studies. Welcome.
Good morning, everyone. Welcome to sunny South Carolina. Okay, so my name is John Pearce. I'm an assistant professor in environmental health at the Medical University of South Carolina, and I'm going to present a framework we've been applying in environmental mixture studies, and in this example, for studies of the exposome. Our framework, we're calling exposure continuum mapping. So at the end of the day, what we are really trying to do here is impose geographic thinking to address the problem of the exposome. And so here we ultimately want to spatialize the problem and use things like spatially correlated learning and spatial pooling to better address complex associations within these difficult data sets, and also use maps as a means to provide this data visualization and location as a proxy to assess risk. So an exposure continuum map is effectively a spatially organized exposure model. Here, the surface of the map represents a continuous sequence of exposure profiles where the profiles are spatially organized. And that organization results in profiles that are close to one another on the map being more similar, and those profiles that are more distal being more distinct. And then we use those spatial coordinates to explore health effects. Application of ECM involves two stages. The first step is the exposure continuum mapping, or the construction of the map. To achieve this, we apply an algorithm known as the self-organizing map, which was introduced by Chadeau-Hyam in the early 90s. Here, again, this is a machine learning style clustering algorithm that looks at the data and identifies profiles. It does it in a way where it uses neighboring information to identify these profiles, and the end result is an organized feature map. We then take the coordinate assignments from this organized feature map and treat it as a spatial surface within the framework of a generalized additive model. And so what this allows us to do is smooth across neighboring locations in order to look at an overall joint dose response function to the total exposome. So we're going to illustrate this by looking at associations between the prenatal exposure data provided by the workshop and our outcome of interest here, phenotype, is birth weight. So very quickly, we're looking at just over 1,300 mother-child pairs. Looks like the data was simulated from 2003 to 2009 across multiple sites. The analytic data we're going to take advantage of here was the exposome data set. We subset this data to look specifically at prenatal exposures. And for this example, we decided to only look at the continuous exposures. And so this ultimately resulted in 50 chemical exposures and 26 outdoor exposures. We have a standard range of covariates that we're going to adjust for when we're estimating our health effects, things like cohort site, maternal age and education, body mass index, etc. And then again, to reiterate, our outcome of interest here is birth weight. As kind of a preliminary analysis, we wanted to run single pollutant models to make sure things were in order. Overall, we found very few significant associations in this type of EWAS type approach. And of course, adjusting for multiple comparisons, we found no significant associations, but this gives you an idea of what our base model looks like. So again, covariates that we're tackling are the cohort, age, education, nativity, parity, BMI, and child sex. So we did find a few significant associations, a few metals, a few of the flame retardants, and it looks like phthalates, as well as cigarette smoking. Our next step was to take the exposure data and construct our exposure continuum map. So to do this, the first step, we standardize our data as common with many machine learning or dimension reduction types approaches here. We then take this standardized data and we try to identify an appropriate number of patterns or size of the map for the algorithm to fit. We have a variety of different metrics that we use or criteria to help us make this decision. In this example, we're going to base it on a kind of an AIC criterion that's often applied in k-means clustering type approaches. So we identify our map size using this criteria. The next step, we fine tune our map in final construction. This looks at optimal initializations and learning parameters for the algorithm. And then we visualize our map here, wanting to essentially illustrate the patterns that are discovered, and we want to label the features for interpretation. So the end result here is what we are framing as an exposure continuum map. So here you have a collection of discovered exposure profiles in the data. These profiles are similar to cluster centroids. They do reflect feature patterns discovered in the data. Here we're illustrating the patterns using normalized radial bars. Here, where effectively larger bar sizes reflect higher concentrations or values for those exposures. Each one of the exposure variables is color-coded, and so as you can see, for example, starting here in the upper left-hand corner, we see our metals data moving all the way through more of our chemical exposures, moving into some of our outdoor exposures here, such as the air pollution measures and the built environment exposures. And so we get a full range of patterns discovered in the data, and the proximity of these patterns to one another on the map reflects their similarity. So for example, these two patterns here would be more similar statistically in terms of statistical distance than these patterns. The spatial distribution or the organization of the mapping tends to place the higher values or the extremes on the extremities of the map and lower to modest concentration values near the center of the map. It's also important to note that we've added a coordinate system here for the mapping. Here is the U-axis or the U-coordinate, which might be framed as an x-axis, and here is the V-coordinate or the V-axis, which will be important in some of the next few illustrations. Okay, so the map can be a very, very important tool for exposure characterization. Part of our overall objective is to be able to really understand the exposome among study populations of interest. And we think that the map really affords lots of different opportunities to help visualize interesting patterns in the data. So for example, frequency distributions, how often these patterns are occurring within your study population is often a very, very interesting question or important question in exposure assessment. And so here we can see the actual counts assigned to our profiles. And so what we see here is that the most common exposure pattern experienced within these cohorts was this particular pattern here in the upper left-hand corner. We can always also look at how population subgroups are distributed across the map. So for example, we know that there were strong cohort effects in the exposure data for this challenge. And so here what we've done is actually mapped which cohorts, the participant cohort that was assigned under each profile. And so for example, here we see that cohort 3 tends to dominate the upper left-hand corner. Cohort 2 tends to dominate the lower left-hand corner. Cohort 4 in the upper right, and cohort 6 and 5 here on the mapping. And what's interesting to note here is that the mapping did not take this information into account at all in its development, and we still see that these exposures, these groupings are well organized by cohort. Another important aspect of these exposure profiles is to look at measures of error or uncertainty in those profile assignments. We can do this here looking at a within class spread among participants assigned to each profile. And another interesting aspect and in terms of understanding the exposures are trying to identify which exposures are most similar and which are most distinct from the other exposure scenarios or profiles discovered in the data. To do this, you can look at neighborhood distances among the profiles. This is sometimes also called a U-matrix in the world of self-organized mapping. So at the end of the day, lots of useful things you can use the map for in terms of understanding the distribution of these complex exposures amongst your study populations. Another aspect that's often used is to look at mappings for the individual components. These are sometimes called component plane maps. And so here, effectively, what you have are the median values among participants assigned to each profile. And so, for example, you can see that some of these exposure planes are very well organized. So for example, if we look at nitrogen dioxide, we see here that the highest concentrations are in the upper left-hand corner of the mapping, and the lowest concentrations are in the bottom right-hand corner of the mapping. And so again, this is in line with the organizational principles of the mapping. We saw this for a lot of the outdoor type exposures. The chemical exposures were certainly different. For example, you see that for some of these chemical exposures, everyone across the cohort had relatively high concentrations, except for the rare occurrence of a few low exposures. We did see some grouping, for example, here with BPA exposures amongst these profiles. For example, on the bottom of the mapping, we see some of the higher concentrations for PPA in this study population. So again, a lot of information here, but what's nice about this visual arrangement is that it allows you to quickly visually assess the distribution of all these features across your population of interest. And so we think this can be a pretty helpful tool, again, in trying to understand how these complex exposures occur in our study populations.
We need to wrap up.
Okay, so the next step is to use this information as a means to better understand health effects. And so to do this, we basically take the spatial coordinates and apply them with a generalized additive model to construct a smooth joint dose response function to variation across the entire exposome mixture. So here what we found in our application were that the lower birth weights, after adjusting for covariates associated with locations in the upper left-hand corner of the mapping, and higher birth weights associated with locations here in the bottom right-hand of the mapping, again, also along this axis here. And there were some distinguishments amongst the exposures in these areas. So in the upper left-hand corner, we saw higher air pollution, pesticide, and measures of urbanicity. In the bottom right-hand corner, we saw distinctions of climate, hotter, drier climate with elevated PM10 or dust, with relatively few peaks in chemical exposures, although some were high. Very quickly, we know that the exposome is very difficult. We think ECM provides an attractive framework for visualization and exploration of health effects. Of course, there are limitations. We know that interpretation of the total exposome is very difficult, so we want to integrate variable selection and important measures to help tease out important components. If you're interested in learning more, we do have all of the code here posted on my GitHub page, and we have a publication under review right now at Environmental Research. I would like to thank my collaborators and funding very quickly. All right, thank you.
Thank you very much, John. That's very interesting. I have a question from the audience.
Okay.
It says, thanks, very interesting work. Is the method works on actual exposure surface as raster grids or for maps, or just mapping based on points of the participant location? How do you account for spatial dependencies among these exposure sets? Couple of questions. I think maybe we can take.
Right. So it's a good question. Right. And so what we're actually wanting to leverage these spatial dependencies through the spatial smooth. So now, we're not, looking at a single exposure pattern. I mean, we're ultimately trying to find an exposure pattern that elevates risk, but we're wanting to leverage the power of neighboring groups. And we need to do that because when we construct these big maps, sample size becomes an issue, right? So to really well-define exposures using an enormous amount of variables, you need a lot of profiles to do this well. And this ultimately results in lower sample sizes, or each individual becoming more unique. And we seek to order them using the mapping and then actually leverage those spatial properties and exploration of health effects.
Thank you. We have another itchy question on categorical exposures that I also want to know the answer, but if you can type it and we can move to the next one. So thank you very much.
Okay,
John,
Paula, thank you so much.
Yeah, very good work. Okay, so we are moving to the next talk. I'm introducing Fei Zhu from the University of North Carolina in Chapel Hill. Fei is going to talk about the Deep-Exposome, a predictive and interpretative deep neural network. So the floor is yours, Fei. I think we can hear you. Fei?
Okay, yeah.
You're there. Okay, perfect.
Okay, so First, I'd like to thank the event organizers to offer us the opportunity to present our research here. So today I'm going to introduce two statistical approaches developed by our research group for the Exposome challenge data. So the first method is a deep learning-based predictive model. The second one is a permutation-based feature importance test developed for black box algorithms. So after the introduction of the two approaches, I'll present some analysis results on the exposome data, and I will close the presentation with few remarks. Deep neural network, or DNN, is frequently applied in biomedical research and is becoming one of the most popular machine learning tools due to its high flexibility in approximating any complex function. So in our proposed research to model the relationship between the X variables and the outcome Y, so instead of directly model this F function parametrically, we employ a feed-forward DNN illustrated in this figure to approximate this function F. Okay, so according to the universal approximation theory, a feed-forward DNN with finite hidden units and at least one hidden layer actually can approximate any continuous function on a closed and bounded subset of a Euclidean space. Though DNN models are flexible, actually there are two big challenges associated actually with how to build DNN models, and also like how to interpret results from DNN models in practice. So the first challenge is that in DNN models, total number of parameters is often substantially larger than total sample size, leading to unstable DNNs. So to solve the issues, we come out with two solutions. The first one actually is a common solution that we use bagging. So bagging has been successfully used for improving unstable procedures, such as classification and regression trees, and also neural network. So the next solution actually is neural network ensembles. It has been argued that many could be better than all, motivating us a novel like a filtering approach for removing low performing DNNs from the final assembled model. I will discuss this procedure on the next slide. So another challenge I just mentioned is that how to interpret DNN models, since feature importance assessment in DNNs actually is quite difficult given their black box natures, motivating the development of our perm-fit procedure. So the filtering procedure actually is based on the following performance scores that we proposed. So the top one is for continuous outcomes, and the second one is for binary outcomes. So according to the performance scores, we can rank bagged DNNs. So instead of using all bagged DNNs, we only use top performing DNNs that pass a given threshold for the final assembled model. So details of this approach can be found in our 2019 biometrics paper, and the software is also available on GitHub at this website. Next on perm-fit, which is a permutation-based feature importance test. So in practice, like feature importance plays very important role actually for model interpretation, biomarker identification, and also prediction improvement. So there are several existing methods for evaluating feature importance. which are listed here. However, those methods actually has a big drawback, and that is either those methods that do not perform like formal statistical inference, or if they do provide like a statistical inference, typically they are very sensitive to like the high collinearity among features. So to overcome that issue, we propose this perform fit algorithm. So the central idea behind this algorithm is to empirically evaluate this importance score. Okay, so we have shown that perform-fit provide statistical valid inference by using cross-fitting and bootstrap aggregating to overcome potential model overfitting. It is also computationally efficient, and since it uses empirical permutation, so to avoid the need of model refit. It is universally applicable to various types of black box models, including random forest, DNN, and support vector machine. And more important, actually, this method is more robust than the existing methods in the presence of high correlated features. And it can provide much improved feature ranking than those methods. So this research has recently been accepted by Nature Communications, and the software and the paper are available at this website again. So next we apply the methods to the exposome data, and we analyzed 5 outcomes: birth weight, BMI, IQ, behavioral score, and asthma. The first 4 are continuous outcomes, and the last one is a binary outcome. So for all of the outcomes except birth weight, our predictors include 220 exposome variables plus some demographic variables. For the birth weight, actually, we only used like 88 prenatal exposure variables plus some demographic variables. So we evaluated 4 models: DNN, Nassau, support vector machine, and the random forest. And the cross-validated accuracy will be used to evaluate those methods, and each method will run with all available features or with top 50 features that are provided by perform-fit. So here are the summary results. So for continuous variables, we report the mean square errors on the scaled outcome, and for the binary asthma outcome, we reported ROC, AUC, and PR AUC in this table. So for the top numbers, the smaller, the better, and for the bottom one, the higher, the better. Okay, so I highlighted the top performing methods, and here you can see that our DNN model either perform the best or perform nearly as good as the top method. And also like highlight two outcomes here, like BMI and asthma. And it turns out that using the top selected features actually can improve the model fit slightly. Okay. And here are the top features, like top 5 features from our DNN models for asthma and the BMI. And interestingly, like when I do the Google search, all those variables have been reported in literature that are associated either with asthma or BMI. Okay, so basically that's all. And some remarks here. Here we have presented Ensemble DNN, which can be a potential like a promising tool for exposure research. And we also presented perform-fit, a computationally very efficient algorithm actually for assessing feature importance. So just like all the other presentations, like for those outcomes, actually we don't identify many significant features And one potential explanation is that maybe those features are poly-risk factors with mixture effects, suggesting that each with us only have a small effect, but jointly maybe they become important. So another potential, another reason is that in this analysis, we only focus on exposome data, and it's very likely that we have missed the important biomarkers and features in other types of data. So our next plan is to perform integrated analysis with, for example, metabolomics and the transcriptomic data. And actually for BMI, we have performed some preliminary analysis and have found that serum metabolomics actually can substantially improve the performance of the ENN models. It can cut the mean square error actually by at least 30%. So that's kind of promising. So here are the literatures, and I highlighted the two papers that are related to this talk. And this is, of course, a collaborative research, and Xin-Lei Li. Xin-Lei Mi actually is my former PhD student who is now an assistant professor at Northwestern University. He and Baimin Zhou, my colleague at UNC, actually have done most of the analysis presented here. And this research is funded by UNC Superfund Research Program and the UNC Center for Environmental Health and Susceptibility. Okay, I'll stop here.
Thank you very much. I wonder whether there are some questions from the audience. And we have Xavier. Do you have tools to explore the marginal associations of some of the selected exposures?
So far the tool is only assess the significance of the feature. Yeah, definitely we're thinking about actually to extend the method to also evaluate the direction of the effect. But keep in mind that the feature may interact with many other features. So
exactly.
Yeah, so marginally they may not show any result effect at all. Yeah.
But the features that come out from PermFit, they may be highly correlated, right?
Potentially, yeah. But the method actually is quite robust to the collinearity among the features. Typically, you'll pick the true signal actually among those correlated features.
Perfect. Okay. Thank you very much. And we move to the next speaker. From our institution, from LIRIS-ISGlobal, as well as from LIRIS, is Jean-Baptiste Guimbaud, and he's going to share with us his work on leveraging machine learning and explainable AI to better understand exposomic data. So a bit more of explanation to these convoluted models.
Hello, everybody. Thank you, Paula. So my name is Jean-Baptiste, and I'm currently a data scientist at Mersens, which is a French company that tries to assess the exposome. And I'm also about to start a PhD on the exposome. So this work was a preliminary work to that PhD, and it was supervised by Léa Maître, which is a researcher at LIRIS-ISGlobal, and also Rémi Casabet and Marc, which are researchers at the LIRIS, which is a French laboratory. Now I will quickly tell you more about the context of its work. So the exposome research field is challenging because of the high number of different factors to take into account in order to model the health. These factors are really diverse from the environmental exposures to the physical activities or so on, and there is a need to take into account the combined effect of those factors. And for now, the most often used techniques in the field to model the exposome are linear techniques like linear regressions, lasso or elastic nets, and also Bayesian methods, which I'm not an expert about, and some dimension reduction and feature selection techniques. And these methods are great, but their expressive power remains limited on to captures complex relationships. That's why I wanted to explore more advanced machine learning methods that can access these complex relationships, non-linear or really complex. And we wanted to compare the results with classic methods that are used in the field, and then to apply some explainable AI method on these methods, which are black box, to get some insights about how our models were getting their predictions. And the last one isn't a real objective, it was more like a curiosity. We wanted to test how feature reduction actually impacts each of our models. So here is a summary of the pipeline that we used on this work. So first, we used the data from 3 tables, exposome, covariates, and phenotype, for a total of 264 features and only 1,300 samples. And from here, it is a classical machine learning pipeline with preprocessing and one-hot encoding for categorical features that we can use with our methods, and also standardization for the model that needs it. And we tried also dimensionality reduction just to see whether it improves or not the prediction of our models. And for classification categorical outcomes, with the problem of class unbalanced, we use oversampling techniques. And finally, we trained our model, which were first a multi-layer perceptron, which is a deep neural network, and then random forest and XGBoost, which are ensemble techniques, and finally a support vector machine. And we compared all of those models with an elastic net, which is a linear regression method with penalization. And to evaluate that, we used cross-validation. And finally, for the best models, we use an explainable AI method, which is called SHAP, to get insights about how our models make their predictions. So SHAP stands for Shapley Additive Explanation, and it's again a theoretic approach based on Shapley value to explain the output of any machine learning model. So it is model agnostic, and basically for a particular instance, it computes a Shapley value for each of the input features of a model. And this Shapley value tells us the participation of given feature to the outcome of the model. And the sum of all of these Shapley values is always equals to the predicted value of the model, so the mean, and the minus the actual predicted value of the model. So this is how it works, and it is actually a powerful technique because it can give us local explanations for each instances, but also global explanation if, for instance, we compute the mean of all the Shapley value for each individual features. But I will tell more about that later. Now I will present you the results. And for the sake of time, I will only discuss the results with continuous outcomes, but there is also some categorical ones. So here we've got an outcome by column. So we train our model on those outcomes and compute the mean absolute error for each of those outcomes, which are the IQ of the children, the body mass index, the birth weight, and the behavior. And for our models, we actually compare them with two different models, which are the elastic net, which is, like I said, an often used linear regression model, and also a naive model, which just computes the mean of the outcomes. And this model is actually useful because it tells us whether our models can get some valuable information from the data in order to predict the outcome, because there is no point to use an explainable AI technique on a model that did worse than this model. So here there is two things to note. First, our prediction weren't really great on most of the outcomes, so they are difficult to predict. For the IQ, it is okay, but the other not really. And the other thing is that the elastic net actually performs very well compared to the other techniques, which was surprising for me. But we managed to get slightly better results, but not really that significant for some outcomes. So now I will focus more on the IQ of the child, because it is the outcome for which we get the better results. And this is the plot of predicted versus measured with random forest, which were the best model that we got. And we can see here that it tends to follow the diagonal. So this means that the model actually were able to get some valuable information from the data. And also we can see here that it tends to overestimate the lower values. And it is maybe because there isn't much of those values in the dataset. And now I will discuss some global explanation that we've got with SHAP. So the first plot is obtained by computing the mean of all Shapley value for each features. And we can see here that to predict the IQ with the random forest, the most important feature was by far the age of the children, and followed by the PFOS, which is a pollutant and an organic food feature. And I also grouped together the features that were really redundant. And we can see here that the model actually tends to ignore the redundant features, which is good. And the second plot show you basically the same information, but with the addition of the direction of association, which can be interesting, and also the distribution of the Shapley values. And here we'll focus more on the age feature, which was the most important features. And we can see, so on the x-axis, it is the actual value of the age. On the y is for the Shapley value. And we can see here that there is clearly a nonlinear relationship between the age and the IQ. So for the children from age 6 to 8, their IQ will improve very quickly. And above 8, there is kind of a plateau. And also for the children with exactly the same age, here we can see that we can get different Shapley values, which is a sign of interaction effects with the age and other features. So on the color axis, I have plotted the value of the PFOS, which is a pollutant. And we can see here that for high value of this PFOS, on the children with the same age, we've got a lower Shapley value, and vice versa.
We need to wrap up.
Okay, so I will skip that. And have to conclude. So linear regression method, we saw that there are still good choices to model exposomic data, at least on this data, because I must also say that the data that we used for that work were partially generated. So we don't know how it was generated or what data were, but it may be induced bias when we try to compare nonlinear on linear models. And the more advanced technique that we used gave us slightly better results, but that was at the cost of model interpretability. So there is a trade-off, and we need to use explainable AI in order to be able to get some insights about those models. But I think that this method can also be interesting for already interpretable models, because it can also show us some interaction effects between the features. And here, for the perspective, so it was just preliminary work for my PhD. So I will probably improve it in the future. And to do that, I could do some things that I listed here, like test also the performances with the omic data to see whether it improved or not the prediction. And yeah, that's it. Thank you very much for your attention, and I will answer your question now. But if you still got some after that, you can mail me at jean-baptiste@merces.com. Thank you very much.
Thank you very much, Jean-Baptiste. We have a question from Lorenzo Fabri. Did you compare the results of SHAP with the standard feature importance for random forest?
Yes, I did, and it was the same, but with SHAP you can also see more precise things like the direction of association, or you can also look at individual feature to see the relationship with the outcome more precisely. So, but yeah, it was the same things. I didn't go too much to the details, but it was the same order at least.
Okay, another question from Lorenzo is: did you try to oversample the small values of IQ?
Sorry, to what?
Did you try to oversample the small values of IQ?
Yeah, not for the IQ, just for the, yeah. I did oversampling, but just for the categorical outcome. But that's a good point. I could try also to do that.
I do have one question. So if two variables are very highly correlated, the Shapley value is going to be a bit random if it's attributed to the first variable, or the variance is attributed to the second, or it's half and half. Is there any rule for that from your experience that will come up? Because that may differ for results with random forests.
Okay, so in that case, you can use clustering that I done here. Comes with SHAP that computes the redundancy with different features, and where it is here. And so it is not really correlation. It is computed with random. No, it was XGBoosts, and so it computes. I think number of XGBoost models equals to the number of features, and it computes it on pairwise features to see whether it is redundant. Okay. And then you can group together these features and see the effect on the models.
Additive explanation. Thank you so much. Yep. Any other question? We have one more. Okay. Perfect. Thank you so much. We move to the next speaker. Next presenter is Alejandro Caceres, also from LIRIS-ISGlobal. He's going to present his work on using causal random forest to determine exposure environments with high sexual dimorphisms.
Hello. Cool. Yeah. Can I be heard? Can I be?
Yeah.
And can you see my screen as well?
We can.
Okay, perfect.
Yours.
Thank you very much. I am Alejandro Caceres, and I am very excited to share with you the results that we got from this data challenge. And in this data challenge, we had asked the question of whether we could identify exposure environments with high sexual dimorphism in human development. So now remember that sexual dimorphism is just basically the difference in an outcome or a trait between sexes. We know that boys and girls develop differently. Okay, their immune response to infections differ from an early age. Their brains grow at different rates, and the prevalence of numerous common diseases like obesity is different. We know in particular that boys have higher risk to obesity than girls. Okay, so why should we study sex differences? Well, for a start, the main difference between people is sex, and this underlies differences in disease risk and outcome. And we also know that precision public health and medicine aim to apply the best intervention at the right time to the right population. And of course, if the main difference between people is sex, then precision public health cannot do without sex differences. Now, sex differences in disease can also inform on etiology. And this is particularly true for age-related diseases like cancer, Alzheimer's, and autoimmune diseases, which are strongly influenced by sex hormones. Okay, and now studying pregenes, like in the data challenge, is very important because this can inform on non-hormonal mechanisms of sexual dimorphism in disease. So our hypothesis was that a co-occurrence of environmental conditions can be identified from exposomic data such that the differences in BMI between boys and girls is highest. So how do we do that? So let's start with the exposome. And the exposome is nothing but a high dimensional exposure data that over-specifies each individual. That means that everyone is characterized by their particular exposome. So of course we want to link it with differences in with BMI differences between boys and girls. So what we do is to propose to estimate sexual dimorphism, that is the difference in BMI between boys and girls, as a causal inference problem. So imagine here that we have a girl with a particular exposome and a BMI, and we ask what her BMI would be if instead of a girl, she would be a boy, but the exposome would be exactly the same. If we knew this, we could do this difference between BMIs, and that'll give us the sex effect given P. So you can use different causal inference approaches. We propose to use random causal forest. And in random causal forest, what you do is that you randomly select a set of individuals in the train set, and then you then select a random set of exposures, and then you run regression trees such that the difference in BMI between boys and girls is maximum in each leaf, right? And then what happens if you have an individual who didn't take part on growing the forest, a test individual. So you take that test individual in the forest, and then you try to find what that individual, on which leaf that individual would belong to, and then you can collect the sex effect on that leaf. You can do that across all the trees in the forest in such a way that you can estimate the sex effect with confidence intervals. So I refer you to the seminal paper of Athene. So what we did was to build a software that we call TEF. It's available in GitHub. And basically this package, which is in R, uses random causal forest to estimate transcriptomic groups where the effect of treatment on an outcome is maximum. So for the data challenge, what we did was, instead of using transcriptomic data, we use exposomic data. Instead of treatment, we use sex. For the treatment, we use sex, and for the outcome, BMI. So when we applied TEF, our algorithm, to the data, in the data challenge, what we found is that 46 individuals from 155 in test set had significant negative sexual dimorphism in BMI. So if you look at these individuals here, that'll be the individuals with the confidence interval in green, and that'll be those confidence intervals will be lower than 0. We call them significant. That means that each individual here, given their exposome, they would have a higher probability of having higher BMI in boys than in girls. Okay, and when you do the random causal forest, then you can rank the variables by their importance. Here you cannot see very well, but the most important variable is PM10. Of course, we selected informative exposures, and this is the code that we use. Basically, you format the data, you do the prediction, you draw the confidence intervals, and then you do the plots as I showed you before. Okay, so now we're interested in extracting an exposure environment. So what do we understand by an exposure environment? So imagine that we collect all the individuals with the confidence intervals in green, and then you have the exposures for each of them, and then you create an average exposure, and you can binarize it. Okay, so now you have an exposure environment, a target environment on which you can target individuals and say if the individual would belong to that exposure environment of negative dimorphism. Okay, so we did that, and here I show you an example. You can target obviously independent studies, but we targeted all the individuals in the data set again. And here is a profile of the individuals targeted in the negative dimorphic group. And here you see on the important variables used in the random causal forest. So these individuals will be negatively dimorphic. And what that means is that basically those individuals in the negative group, well, the boys will tend to have higher BMI than the girls. And you don't see that in the neutral dimorphic group. Okay, so this is the code that we use. After you do the prediction, you do the targeting, and that creates you all the plot that I showed you before. And of course, the method also tests the interaction that I just showed you, and then that interaction we confirmed that it was significant. So we detected this highly dimorphic group where boys have higher BMI than girls. So now, of course, now we have a way to classify individuals by their exposure and this exposure environment, and then we ask whether there were molecular correlates of the exposure environment. And then the first thing we did was to run a differential expression analysis, and then we were very surprised to find MET-L3, which is a subunit of the most important methyltransferase of mRNA. Also very importantly, MET-L3 is involved in adipogenesis and M1 macrophage polarization, and obesity is characterized by the inflammation of macrophages. We also know that one of the most important functions of FTO, the fat mass and obesity associated protein, is to reverse exactly this type of methylation. And we also know that this type of methylation regulates chromosome X inactivation in females, which is a common process in females on which the extra chromosome gets silenced. Okay, so we run a enrichment analysis. We found 4 enriched pathways, all of them related to sugar transporter activity. They were led by these 4 genes. All of them were glucose transporters. So that was an interesting finding. And since the finding of methylation of MET-L3 was an important finding, we wanted to see if there were some consistent association of the genes of the glucose transporters in relation to their methylation and expression. And we found a consistent association of the glucose transporter 6 with increased expression of methylation. As the expression of MET-L3 goes up, the methylation of this gene goes down, and as the expression of MET-L3 goes up, the expression of this gene goes up. And here you see the negative dimorphic group in green. So this is a very interesting finding also because the glucose transporter 6 is involved in inflammatory macrophages M1. Okay, so since our data was pointing towards immune response, and we have data on immune cells abundance in the data from the data challenge, then we run association tests between BMI and the interaction between the exposure environment and sex. Okay, and we found that there were significant associations of BMI with monocytes and granulocytes Remember that monocytes are the precursors of macrophages. And for the interaction of sex and the exposure environment, we found a significant association with CD8 T cells. As for the genes that I discussed before, for the glucose transporter 6, we saw a significant association with monocytes, which was interesting. And also for the interaction between metal-3 and sex, we found a high significant association with natural killers. So there is an immune response that is associated with these features that we found. So we run a differential methylation analysis, methylome-wide, and we found 2 CpGs that were significant, and they also fell in genes with immune function. This GNLY and HLA DPB2. So my final remarks, we identify an environment of negative dimorphism in BMI, where boys tend to have a higher BMI than girls, which could be linked to meaningful molecular mechanisms. These include genes involved in immune response and adipogenesis. The 12 exposure environment included increments in PM10, low meteorological temperature, high humidity, and the reduction and increment of specific organochlorines, phthalates, and phenols. The exposure environment should be validated with external data and simplified, standardized for public health interventions. Also, our method is sensitive on variable selection, so we need to look at that. We found that exposure environments of high dimorphism for IQ and asthma, but they could not be linked to molecular function. We did not find an exposure environment of high dimorphism for obesity, but for this environment of negative dimorphism for BMI, we saw that it significantly modulated the obesity dimorphism. So I think our data brings evidence on the more general question of as to which extent inflammation from environmental exposures can trigger obesity. And of course, our approach is through sexual dimorphism. So I want to thank you for your attention. I have put the slides online and the code as well, that you can check all the analysis that I didn't have time to show. So I'm going to share with you the link to that in the chat, and I will leave it there. Thank you.
Thank you very much, Alejandro. I think this talk or this topic highlights something that we usually ignore, which is the effect of sex in medicine, which is a very hot topic now in ethics and the ethical use of AI. So thank you for that, and really interesting results. Do we have questions from the audience? Okay. I'm interested to see whether, if you would expand this, what other topics would you like to look at sexual dimorphism in?
Well, we have already, and then, as I said, this is an application of the exposome. We first started with the application of the transcriptome, and then we saw a similar pattern, but instead of having exposures, we have like a group of highly dimorphic features, but on transcriptome data. And this is interesting because you can also classify individuals, and when you classify individuals on sexual dimorphism in immune response, you can predict better survival in cancer, and also in autoimmune disease. So this is something that we're working on. Exactly.
Did you check out the balance of the data sets and whether there are some biases that you would account for what you are looking at?
No.
Because I think,
well, we didn't have
this is a great result. Fascinating. No, it's just a point that if you actually found these results, the next question will be, okay, just to check that they're not that they are well balanced, so then that your results are, can be shared as valid.
Yes, of course. I mean, the most important thing for us is to classify individuals, and once you have it, you can extrapolate it to independent data. So I think for me it's more pressing that we can translate these to independent studies, and that you have biological correlates of that. then I'll believe everything that I've done before.
Exactly. Anyone else? Another question? Well, thank you so much, Alejandro.
Okay, thank you.
Thank you. And we are moving on to another talk by a team. This is Lucille Broseus and Paulina Jedinak from the Université de Grenoble in the Alps. Welcome.
Hello. Give us a second. Okay, perfect. So I guess you can see the presentation now.
Yes, we can.
Okay, so hello everyone. My name is Paulina. I'm affiliated at the University of Grenoble Alps in France, and together with my colleague Lucille, we will present the results of our search for the most relevant risk factors for childhood overweight and obesity. So the objective of our work was to assess associations between child postnatal exposome and risk of overweight and obesity, which has been shown to be an important factor driving overweight problems later in life that in turn could result in several related health disorders, which has a special interest from the public health field. So in our work, we concentrated, and in our approach, we focused on postnatal personal exposome, which included lifestyle and chemical exposures. And we studied association of these exposures with categorical child BMI, focusing on normal and overweight and obese children. So briefly, first, I would like to give you an overview of the exposure data that we have been dealing with. So we looked at 24 lifestyle exposures, mostly categorical variables, these were, and they most included diet covariates, and the chemical exposome that were 40 exposures from 7 different families, and plus continuum, that was the biomarker of tobacco exposure. So first of all, I would like to present you some challenges that we encountered when we first looked on our data, and that stemmed from the data structure itself. And we think that these challenges are common for most of the studies carried in our field. So just to give you a few examples, for example, for lifestyle exposure, we had to deal with a mix of categorical and continuous covariates. And also some of the covariates, like the diet covariates that you have summarized here in the figure on the right, they were already summarized into tertiles. And in the figure, you can see how unbalanced the structure of some of these variables was in terms of the different BMI categories. And finally, we also noticed that, for example, some diet covariates were aggregates that were kind of ambiguous, because just to give you an example, the consumption of French fries sometimes was categorized as consumption of potatoes, which were in turn classified as vegetable consumption. So this was also potentially biasing our analysis. Another challenge that was already several times discussed in the previous presentations was the structure of the exposure data set, which first of all, there were several strongly correlated exposures, especially within the families such as organochlorine compounds, but also phthalates, PFASs, and so on. And also, despite the fact that there was a log transformation performed prior to the analysis to diminish the effect of outliers, you still can see that even after this transformation, the distributions of several chemicals were still highly heterogeneous, and there were still outliers present. There were also some the issues when we looked on the confounding factors that we adjusted our models for. And for example, if you look on the age of the child, you see first of all, the range of the age was pretty broad. So from 6 to 12 years of age. So for studying metabolism, like metabolic features of a person, this can be pretty confusing because on such a long time span in the childhood, there can be several different features that characterize these individuals. Also, there were clusters so that were connected, linked to the cohorts. So different cohorts had different child age structure. And also another example is the maternal education that, as you can see, most of the participants, they had a higher university diploma, which is rather not representative for the general population. So all this also hinder the generalizability of our results and conclusions. And finally, when we looked at the health outcome itself, so the categorical BMI variable, you can also see that there were several confounding factors for which the structure, the data structure was pretty unbalanced. So on the right, you can see the BMI classes distribution in function of cohort, but also in terms of other covariates, there was a very high, sometimes imbalance in the health outcome structure. So this was a very brief presentation of the nature of the data that we were dealing with. And now I would like to pass the microphone to Lucille, who will present you the tools and methods that we applied to deal with these at least to try to deal with these limitations and to try to identify the risk factors of child overweight and obesity.
Thank you. So to address these challenges and compare the respective effects of lifestyle and chemical exposure, we devised a multi-step approach which combines machine learning, usual regression, and graphical representation. So first, we tried to search for best predictors of child BMI classes using multivariate ordinal random forests. The asset of this method in our setting is that it can be applied to mixed data, that is setting where we have both categorical and continuous data. By including confounding factors in the model, we can somehow adjust for them. Then it allows to rank best predictors using the notion of variable importance. And there are tricks that can help handle unbalanced classes. But once we've got best predictors, we want to have a better interpretation, a better understanding of the results and why we obtain these results. So on whole, predictors that were given by ordinal random forests, we performed univariate ordinal logistic regression to estimate the associated risk of each class and the direction of the effect. And we combine these results with, in the case of categorical data, with multiple correspondence analysis, which is the categorical version of PCA, and which allowed us to spot mixing between lifestyle exposure. And the idea of this multi-step approach was to detect spurious or indirect associations. So here are the results when we applied our methods on the data. So first, as I said, we applied ordinal random forest on both type of exposures, lifestyle and chemical exposures independently, separately. And what we obtain is that the best predictors are actually the continuous covariates. So here, in the case of lifestyle exposure, the best predictors is the only continuous predictor, which is the number of hours of sleep per day. And we've got also other good predicting covariates in the diets family, which are high consumption of bakery products, high consumption of vegetables, and also the class of organic food consumption. On the other side, on chemical exposures, we've got very high discrepancies between variable importance. And for both families, we fixed an arbitrary threshold to select covariates for further interpretation and analysis. So once we perform this analysis, we try univariate ordinal logistic regressions on predictors. And so here for lifestyle exposures, one interesting thing is that in the first place, we find that high bakery product consumption is a good predictor of obesity, but once we have a look at the direction of the effect, we would conclude that eating many bakery products would protect against overweight or obesity. And we can detect also some such surprising conclusions. So to better understand these results, we perform multiple correspondence analysis, which allows to visualize correlations between diets, covariates, and their level. And what we see here, for example, is that here low consumption of bakery products is associated with a high consumption of organic food. And on the opposite, high consumption of bakery products is somewhat correlated with consumption of ready food and with low consumption of ready-made food and low consumption of soda.
Sorry.
So there are a spurious correlation between healthy behavior and unhealthy behavior. On the other side, applying the same method, so based on these results, we can filter somehow and interpret spurious association. But on the other side, on chemicals, we had to just apply the methods and select arbitrarily the best predictors without relying on this interpretation and curation. So once this was done, we combine both lifestyle and chemical predictors in a model. So both in a random forest and in a multivariate ordinal logistic regression. And what we find after curation is that sleep and high consumption of vegetable were associated with lower risk of overweight. But still, the complexity of diet data, for example, the binning into tertiles and ambiguous aggregates prevents from having a very satisfying analysis based on this data.
Yeah. Okay. Lucille?
Yeah. So we also found significant associations between the risk of overweight and chemical compounds, which are known associations actually already highlighted in previous studies. So as a conclusion, we know that socioeconomic factors are known to be major determinants of overweight and obesity. This multi-step approach allowed us to identify several lifestyle and chemical exposure associated with child BMI. But for example, here you see the results of the ordinal random forests, which shows lifestyle exposure, chemical exposures, and or major confounders. And we can see that the predictive values of exposure is not equal if we compare chemicals and lifestyles. Chemical exposure looks to be better than lifestyle exposures as predictors of obesity. But we also see that some chemicals exposures had more predictive power than confounders. And as we noticed that we found surprising associations with lifestyle exposure and that can be understood and curated graphically, something that we cannot do with chemicals. This one warns us about the meaning of associations that we found with chemicals. So probably there are many spurious associations that we cannot interpret. And what could explain the fact that chemicals are much better predictors than lifestyle exposure is that they could be partly surrogates for hidden socioeconomic factors. For example, our best predictor was HCB exposure, which is known to be mostly observed in rural and industrial areas. So thank you very much for your attention.
Thank you so much. We are a bit over time, also stepping into the discussion. So thank you very much, Lucille, for the talk, and Paulina. So if anyone has questions, maybe we can put them in the chat and maybe we will take before the networking and the break, just 10 minutes to bit of a wrap up. And I'd love to hear some comments from the audience. Just maybe my observations from, it was excellent work, really, really great presentations, everyone. I thought that everything, like all the topics addressed were very diverse. Also the themes were very diverse. And one of the things that I point out, maybe for me, it's the relevance of the machine learning explainability aspect, which is the driver of new hypotheses and new ideas. I think for exposome type of related questions, just if we can't explain the predictions, then it's not interesting anymore. So that's why we just don't see black box type of questions, but then we see different algorithms to explain why the observations, and then we draw new hypotheses. I don't know if anyone would like to comment on this or other topics related to the talks, and please don't be shy and just step in, and so we can have a discussion. Any takers? Any of the panelists?
Well, maybe I'd like to say something about it. So I think that for me, my approach in this case was to formulate the biological question first, and then try to use the algorithm to answer that biological question. So the interpretability is at the beginning of it, and then you use the machinery that you need for answering the question, and then that would lead you to other things. So maybe if we ask the right question at the moment, at the beginning, then things will come easier instead of trying to figure out significant results or things that we don't know how to interpret. Of course, when you want to classify individuals, you want performance, and then that's a different issue, I suppose.
I think your point goes to supervised versus unsupervised. When you have the question, then you can use a more supervised model, but then also unsupervised will help you. But So coming from biomedical data science, and my background is not so much related to exposome actually, I find that exploratory analysis usually don't conduct to very definite answers. They are very useful because then you don't know where you could find, but when you have the question, machine learning gets better answers when you have a very, very well-defined and planned study. I don't know, maybe we can initiate a debate on that. And people that have done exploratory analysis, what's your experience doing those? And whether you found that you were scrambling in the data or that you actually were getting something out of the models. So we have Carl. Yes, machine learning can do both, but please, maybe you want to discuss about your experience working on different topics. This is just a personal experience, right? Like what I have done in different projects, mostly very, very time sensitive projects when you have to deliver an answer. So would you like to, I don't know if you can talk, like, can you put the audio? I think maybe only the panelists are able to discuss, and we can move on to the discussion room.
Yeah, I was actually added as a panelist. So I think there is actually a misconception that supervised unsupervised corresponds to confirmatory versus exploratory, and I think that is a misconception. I think that you can use a combination of supervised and unsupervised methodologies to be either exploratory or confirmatory. So you can mix and match any way you want to go. And Alejandro's idea to start with a hypothesis, I can do that. But I can also go fully exploratory, and I hope to show that in my presentation, which will be after the break and the whatnot, that we can start without having hypothesis of what is the exposure of interest, what is the outcome of interest. And we use a combination of methods to fish out those hypotheses, drill down deeper into it. But of course, then that needs to be confirmed. And then the confirmation comes from either using other cohorts and similar methodology, or using other methods or other types of data. So various types of triangulation of methods, triangulation of using different observations. But to not say that these methods are useful only for this or only for that. Any method is useful for basically any purpose.
No, there I am with you. I was more thinking about the classification of questions, but for me, the times that I've applied like a driving question and then apply the methodology was more like directed to objective. But I'm with you that you can actually do anything, and actually you can start with a question and finish with another question, and your initial question was never answered, right? So I'm really looking forward to your talk, whether you started with more exploratory approach and then try to use other topics. I very much look forward to that. This is just my experience, right? When you have like to deliver in 2 weeks or 3 weeks, going to an exploratory approach may be a bit stressful, but I'm looking forward to looking at your talk.
Thank you.
Okay, I think we will move to the networking space. And I wanted to close the session and really appreciate all the speakers, the presenters, and the organizers for participating in this session. Thank you so much.
Thank you, Paula, for chairing this session.
Yes, Paula, thank you. Great session.
To the chat rooms. Hola, Elia. Hello.
So welcome back, everyone. We had a shorter break than expected, and some actually took advantage of the special chat to have more conversation. I hope you're still fresh enough to follow the last topic of the day. So for this topic, we actually, like from the organization committee, we were struggling a bit to split the abstract. We're not sure like where to put different abstracts integrating omics in the analysis. And we thought that there was one set of abstract was trying to put more the omics as a triangulation tool, trying to extract more causality. So that's was inspiration for this topic. And we're going to start with Carl Brunus from Chalmers University of Technology, who is going to present about the omics models for Exposome Health Association. For all the speakers, you have 12 minutes, and I will give you a reminder if you over time. The floor is yours, Carl.
Thanks a lot, and thanks for the introduction and the possibility to talk about our stuff. I hope you hear me, and I hope you see my presentation as well. Just please let me know.
Yeah, we can hear you and see your slides. All good.
Awesome. So it's obviously impossible to talk about everything that we've done in 12 minutes, but I'll give it my best shot. So I'm group leader of computational metabolomics in our larger research environment on food and health. And I'm just some shameless self-promotion. Our tasks fall into roughly 3 categories. It's a metabolomics data generation, where we generate roughly 10,000 untargeted LC-MS samples for metabolomics. So it's kind of large-scale operation and facilitating that. Omics data analysis with a clear emphasis on machine learning methods and focusing on reducing bias and performing especially variable selection with minimum bias, really, and not focusing so much on single grand unifying theories, one method to rule them all, but actually building data analysis architectures, combining different methods to perform more advanced tasks. And that is what we use when we look at associations between exposures and health through omics data. So we try to develop sort of machine learning based molecular epidemiological approaches where we can integrate multiple exposures and multiple outcomes using a sort of meet in the middle approach. And we use that predominantly as a tool for hypothesis generation and exploratory research. So that being said, I have had the luxury of sort of supervising this team forward, and all the work's been done by these 3 amazing people, 2 PhD students, Elise and Tessa, and a postdoc, Anton. Me and Tessa, we developed and applied some of these approaches for looking at association between PFAS and type 2 diabetes, and now other outcomes and exposures as well. So we wanted to do a very agnostic approach to not tell the data what hypothesis we have, but actually start totally wild and crazy, answer all of the challenges at once, find the most important things, and just have a lot of fun. So we have heard about computational efficiency. We are not computationally efficient. If you calculate the sum of CPU hours going into what we have done, we are measuring in the thousands. So it's a lot of heavy machine learning, but fortunately we have computers to do that for us. So that being said, our framework for integration of stuff is, as I said, a meet in the middle approach. So if we want to make epidemiological modeling of exposures versus outcomes and want to make that explicitly using standard epidemiological tools, it becomes very complex very fast. So instead, we see this as a variable selection process. We look at the omics profiles that relate both to the exposures and to the outcomes, the phenotypes. And then having selected that part of the omics data, we can pretty much integrate anything we want into that. Hopefully you'll agree. But one thing that we have not considered due to the lack of time, because we have not used any domain knowledge. We don't know this helix data, so we don't have that advantage. So we know that there are alternative causal hypotheses, for example, using the omics variables as markers of symptoms rather than being on the mediating pathway from exposure to outcome. The core methods that we're using, so the bulk of the CPU hours goes into this algorithm, mover, multivariate modeling with unbiased variable selection in R. Basically, it's a random forest, but that's performed within a repeated double cross-validation, which makes everything computationally costly. But we, on top of that, add insult to injury, we perform recursive feature elimination within the double cross-validation. So this requires modeling and modeling and modeling and modeling, but the results of this are that predictions and variable selections become virtually unbiased. So we're very happy about that and tend to use supercomputers a lot to not overburden our own machines. But to adjust for covariates, we then subject selected variables to covariates using partial correlations or GLM type of models. We do some latent variable analysis to aggregate variables into modules or clusters or whatever you want to call them. And then we visualize everything. We put all of this together into one tool, which we call the triplot, which is basically a glorified PCA where we can stack different information layers to make sense of this. So as I mentioned previously, we focus on analytical pipelines or data analytical architectures. So the entire workflow that we're seeing here, this represents everything that we've done. On the left-hand side, you'll see that there is a selection of exposure variables. Here we find the selection of omics variables related to those exposures, and here those variables, omics variables that also associate to the outcomes of interest, and then we aggregate everything in the triplot. So we'll look at this in slightly more detail. So we have 6 phenotypes that were given to us in the data set, 2 categorical, 4 continuous. And then we had 222 exposures that we could address efficiently as continuous variables. Of those, only 2 of the phenotypes could be modeled with a Q-square of above 0.2, which is an arbitrary cutoff, but we needed to just use some kind of arbitrary cutoff for the sake of time. And those were ZBMI and Raven score. And the variable selection of the exposures, we found 106 unique exposure variables. And then we adjusted those for cohort, age, and sex individually using partial correlation against the phenotype variable of interest. And that led us to a selection of 26 exposure variables, these guys. And you'll find PCBs and PFAS and some other guys here. We take these 26 exposure variables, they become in our next stage of mover modeling, and this is where the bulk of the CPU hours come into play. Those are the Y variables, and now we have all of the omics data sets as our predictor variables. So we did it omic by omic, and also combining into multi-omics. And the core modeling here is random forest within this repeated double cross-validation. So we don't do any weighing of variables. It's a very naive integration of all the variables together in multi-omics. And we trust the variable selection internally to figure out what are the most important variables. So what we find is that from the single omics, the serum metabolome is by far the most informative one, although the number of metabolites is relatively low. And gene expression, methylation, urine metabolome, the very few exposures get selected. But in the multi-omics, that also translates into the variables of interest, that methylation, 0 methylation variables actually survive this pipeline. But when we do the multi-omics, we find that more models become significant or interesting with these exposures now going from 26 down to 11. And if we break down the 244 selected variables from this multi-omics model, we find that methylation and gene expression from being 5 and 0 become 44 and 165. indicating that there's a lot of crosstalk between the different omics layers. And methylation, while not super strong in itself, has complementary information content to the other omics layers. Then we do GLM versus the two outcomes that were selected from us because they produce relevant models, BMI and Raven, altogether 57 unique omics variables were selected, some overlap between BMI and Raven, and we see again that methylation survived, but serum is like really, really informative. So we put everything together in the triplot, which is basically a PCA of those 57 omics variables, and the lower loadings, they are grayed out, but they are still there, obviously. So there are two components here that we have focused on for time constraints. On top of this PCA, we can put partial correlation of the scores with the exposures, and then we adjust those for cohort, sex, and age. So we find that in the first component, they associate with PFAS and organic chlorines, and the second component associates inversely with organic chlorines predominantly. But we can also take the scores and associate them to the outcomes of interest, and then we see that the first component relates to Raven score, and the second component relates to BMI. This is then adjusted for cohort, sex, and age. We also took the liberty to further adjust them for additional factors, and we see that these beta coefficients for the component association to these phenotypes is very, very stable. So there's some a metabolic regulation or metabolic profile pattern that associates with PFAS and organochlorines and Raven, and a pattern here associating with BMI and inversely with those organic chlorines. So we can dive deeper into this, into BMI and IQ. And if we look at the literature for PCBs and metabolites, there's very strong literature support for this, as well as for omics variables and ZBMI, as well as the direct one. So basically what this tells us is that this method, which did not start with clear hypothesis, but it's just a brute force way to throw computer powered data to fish out potential hypotheses, finds what is really known in literature. So that strengthens us in sort of digging the approach. The second one, we find not so much support in humans between these organic pollutants and IQ. So strengthened by these results on the left side, we can say that, well, then this becomes potential targets for future research. So it becomes, as I said, a tool for hypothesis generation. There are obviously some limitation that we do a pre-selection of exposures, and that's just in the interest of time. We know that some exposures have mixed associations to the outcome, both one type of metabolic regulation that can increase risk and one which can decrease risk. So then the overall association is null, and then all of those exposures will be filtered out. But yeah, that's a time constraint. They are sensitive to adjustment. But more importantly, perhaps, is that we are limited by the selection of omics variables. So if there are mechanisms that are not captured by the omics data that we have, then we will not find those biological pathways using this approach. For example, regulations in the mother before and during pregnancy, and also that the coverage of the metabolome and the proteome, they are really, really limited. And obviously general epidemiology, that we're sensitive to confounders. Moreover, obviously that we only investigated 2 omics modules, and several details may be hidden in the smaller components. And given a bit more time, we could have drilled into this more, and we would love to, but time did not allow. Further, we did not investigate alternative causal hypothesis like with this, that omics variables could act primarily as markers of symptoms, but this could easily be modeled, of course, and we could look at the differences to go further with this. And that would be part of mediation analysis that unfortunately we did not have time to go into, but we do not obviously investigate true causality. We would need interventions and like Mendelian randomization for those purposes. But that's again a general limitation, not perhaps specifically of this method. So my take-home messages, is that without using specific domain knowledge, without knowing the data, just throwing computer power and sort of data analytical architecture, we have shown a data-driven approach to generate the sort of most apparent hypotheses for associations between exposures and outcomes using the omics data. We combine machine learning with GLM to manage both multicollinearities and various aspects of modeling complexity. We rely heavily on the unbiasedness of the variable selection that we have. That's why we use this mover tool. And if you're interested in that, I'd be happy to talk more about that. And the meet in the middle approach is based on this triplot tool. And those are published and open source R packages. So we hope to zoom in the most important pathways, or the pathways most easy to find, going from exposure through metabolism to outcome, showcasing that we have biological plausibility, stepping into finding potential targets. And this approach, using the really meet in the middle approach, is a strong potential for generation of hypotheses. And there I have finished a bit over time. Sorry about that. Thank you.
Thank you, Carl. Yeah, very impressive work by you and your students. It's actually interesting how your findings with the different findings when you combine the omics together or when you're taking them individually. Well, since there is no question in the chat, I'll just have one brief question regarding this. Like, actually, how does it work? Why, when you combine, how the omics combine? Like, is a correlation between the different layers taken into account? Like, how...
Well, so from a methodological point of view, we just concatenate all the variables. So we have a really huge data set that we throw into the mover algorithm. But why do they get selected? Well, random forest operates predominantly on the complementarity between variables that they add on to complementarity to information content. And probably those layers, the methylation layers, has some unique information that is not very strong, but unique. So it complements the other ones. So to me, it makes sense. Gene level data is very, very high up in the sort of central dogma in the gene cascade. And what we find on the metabolome, serum metabolome, it's closest to clinical phenotype, and of course that's going to be the strongest omics. But so I think that's what's happening.
Right. Well, thank you very much for your presentation. We will move to the next speaker.
Thank you.
So our next speaker is from Stanford University, Chadeau-Hyam. The floor is yours.
Hello, everyone. Can everyone see my slide and hear me?
Yeah, we can hear you and see your slide. You may want to put it in full screen mode.
Yeah, sure. All right. Okay, it's good now?
Yeah, perfect.
Okay. Good morning, everyone. I'm Xiaotao, and I'm from Stanford University. I'm a postdoc at School of Medicine, and today I want to talk my study on this exposome data. And my topic name is decoding links between the exposome and health outcomes by metabolomics analysis. As we all know that the health outcome, namely the phenotype, they are affected by the exposome and the genomics. And so it is very important that we want to know how exposome can affect the health outcome. And in our recently study, this study has been submitted, and we also found that the exposome can contribute to the phenotypes. Here we just use a blood test and the cytokines as a health outcome or the phenotype. And we found that a lot of phenotypes can be contributed by more than 50% from the exposome. So it means that the exposome can significantly affect the health outcome or the phenotypes. So, and how the exposome can affect the health outcome, because we know that for the in the body or in the cells, we have a lot of molecules, transcriptome, proteomics, and metabolomics, and they can construct a very complex very complex regulatory network. So how exposome can affect the molecules in our body and then affect the health outcomes. So our hypothesis here is that the exposome can affect the phenotypes where the internal omics, namely the transcriptome, proteome, and metabolome. And so here we just use a data set from the exposome challenge data set. Here we just only use a childhood data set. And because for the childhood data set, we have we have the same time point for the exposome and the internal oms. So here for the exposome, we selected the indoor, outdoor, and the chemicals. And for the interomics, we selected the transcriptome, proteome, serum, and the urea metabolome. And for the phenotype, we selected the 3 continuous phenotypes, namely the IQ, neuron behavior, and BMI. So our step 1 of our study, we want to know if the exposome can affect the health outcomes or the phenotypes. And step 2, we want to know if the exposome can affect the internal omics. Here we just use the transcriptomics, proteomics, and metabolomics data from a serum and urine samples. And the third step of our study, we want to know if the exposome affected the phenotype. They just wear the internal omics. So here we just use a very popular and very strong analysis method, the mediation analysis method. So from step 1, we just use a linear mixed model to find the associations between the exposome and the phenotypes. And of course, we also adjusted a lot of the confounding factors provided by the organizers here, the child sex, year of birth, mother BMI, and so on. And here, the left figure just shows the association networks between the phenotype and exposome. And just to make a briefing summary of this network, we can deduct 131 associations in total involving 3 phenotypes and 79 exposome features. And here we just use cutoff p adjusted values less than 0.5. So next, we just want to know the association between the phenotype and exposome distributions, and we can see that most of exposome, they have the associations with 3 phenotypes, and we can see that most of the associations are from the outdoor exposure and the chemicals. And from here, we can also see that the phenotypes, namely the IQ, BMI, and the behaviors, have very similar numbers of associations with exposome. And here we just use this figure to show how many exposome for 3 different phenotypes. And we can see that they have very similar numbers, right? For the IQ, we have 39. Certain exposome features have significant associations with IQ, and for the body BMI we have 40, and for the behavior we have 52 exposome features have a significant association with phenotype. And then we just want to know the overlap between the exposome associations with the 3 different phenotypes, and we can see that the overlapping of the exposome features with 3 different phenotypes is not so big. The overlapping is less than 50%. So it means that for the 3 different phenotypes, they have different exposome features who can affect them. So from this result, we know that exposome has very significant associations with phenotypes, and we have 110 exposome features in total, and around 64.5% of the exposome features, they have very significant associations with the exposome with phenotypes. So next, we just want to know if the exposome can have very strong associations with interomics data. Here we just use the transcriptome, proteome, and metabolome. And from this figure, we can see that we constructed a very complex and has a lot of nodes and edges of the correlation network. And finally, we get this network involving 888 nodes, and around 7,000 edges. So it means that the exposome have very strong association with internal arms data. So we also want to know the nodes and the edges, namely the associations between the exposome and the interomics, and the distribution of the node and the edges. And from the left figure, we can see that there are -1 exposome features involved in this correlation network. And for the interomics, we have 789 interomics features in this network. And from the right figures, we can see that most of the associations between the exposome and the interomics are in the serum metabolome and the outer exposure. Even we know that in the internal omics data, the serum metabolites, the number is not so big. So now we have two results from the previous study work. We know that we have 110 exposome features, and 79 out of the exposome features they are associated with the phenotypes, and 91 out of 110 exposome features associated with the interomics features. And then we just want to know the overlap between the two exposome features, between associations between the exposome, between the phenotypes and the interomics data. And it's very interesting that from this figure we can see that they have very big overlapping. So it means that so the exposome maybe exposome features associated with the phenotype and interomics, they just with the same exposome features. So next we want to know if the exposome they affected the phenotypes where just where the interomics data. So here we just use a very useful and strong analysis method, and this method is called mediation analysis. And this method is very popular in the biomedical analysis field. And here I just issue a paper recently published in the Cells, and they just use this method to find how the microbiome can affect the phenotypes where the plasma metabolites. And they found a lot of and very interesting microbiomes which can affect the phenotypes, and they affected the phenotype just where the plasma metabolites. And so this is method we used in our study. We just use the same method, mediation analysis, to find if the exposome can affect the phenotypes where the internal omics data. And from this figure, this is just example networks of the mediation analysis network. And I just selected the top 250 average causal mediation effects. And the left column is the exposome features, and the center is just the internal omics features from the transcriptome proteomics data and the metabolomics data. And to the right column, just the phenotypes. Here, just I said before, I just use example networks from the top 25. So in fact, we have 3 phenotypes can affected by the exposome where the interomics, but here we just shoot 2, IQ and the behavior. Yeah, so it means that we get a lot of linkages from the meta analysis, and just to make a brief summary from this network, we have 1 solent and 188 mediation linkages. And here we just also use cutoff as 0.05, adjust the p-values. And in this network, we have 3 phenotypes involved in this network and 124 internal omic features. And so we also have 56 exposome features here in this network. So we want to just know how what exposome features can affect the phenotypes. 3 different phenotypes where the internal omic features. So here I just get the separated networks. So this is a network for the IQ, and just like the whole network, the left column is exposome features, and the center is the internal omic features, and the right is just the IQ phenotype. And the color of the edges just mean that the direction of the correlation. So here we can see that the red just mean that the positive correlation, and the blue mean the negative correlations. And I just want to take example of these networks. It's very interesting. We just searched the literature online and found that the PCA lipids have very they have the positive correlation with IQ. And here in our study, we also found that we have 26 lipids in this network, and we found that 25 of the 26 lipids they have the positive correlations with IQ, chart IQ. And we also know that the NVDI, namely the normalized difference graduation index, is positive associated with IQs for urban kids, but the mechanism is not clear. So it's very interesting because we found that the PC they have positive association with the IQs, and the NVDI also can affect it positively affected the IQs. So we just want to know if the NVDI can affect the IQs where the PCs. So here we just show the NVDI how they can just positive affect the IQs. And here we can see there's these triple plot, right? And we can see that the left bottom is the.
Yeah, if we can wrap up, if you can reach
okay.
your conclusion.
Yeah, sure. So we can see that most of the NVDI they can affect the IQs where the PCs, and the average of the contribution from the NVDI to the phenotype is 34%. So it means that the NVDI can affect the IQs where the PCs in our study. So we also have two examples of the exposome to the two different phenotypes, but we have no time here, so we can discuss later. And so make a brief summary of our study, and we just know that exposome is associated with internal omics and photo-associated with phenotypes. And we also found that the internal omics mediate the interactions of exposome and the phenotypes. And finally, I want to thank our exposome teams in our lab, Guimbaud and Dr. Alejandro, and our supervisor, Michael Snyder. Thank you, and I'm happy to take any questions.
Yeah, thank you very much. This is very impressive work, and yeah, I really like how from this data integration you can actually answer some biological questions. I don't know if there's any questions from the chat. I don't see any for now. Maybe I'll ask you, and I'm sorry if you said it at the beginning, but how did you take into account the main covariate, the main confounding factors in the data? For example, we know that a green space axis is very related to the cohort.
I'm sorry, you mean how I adjusted the co-founders?
Yes.
Oh yeah, so here for the associations between the exposome and the internal or the phenotype, I just use a linear mixed model here. So I just use, so here I just put, so I just put all the co-founders provided in the data set,
right?
And yeah, as a covariance in this model, it means that, for example, in the exposome and the phenotypes, right? So the phenotype just as a dependent variables, and the exposome features as a independent variables, and to the child sex, year of birth, and so on as a covariant in this model.
Okay. Very good. Yeah, I really like how you use all the phenotypes. It's interesting to see also the overlap between them, and yeah, very good. I don't know, maybe we can, if there are some questions for you, we can take them for the general discussion.
Okay, thank you.
Well, thank you very much for your presentation.
Thank you. Bye.
We'll move to the next speaker. So this is Kongrong Yang from Assault University. I don't see. I see you are opened your mic.
Yeah, I have to wait until Chateau stop his sharing. Yes, okay. Do you see me and my slides?
I see your slide, I hear you, but you don't have your video on.
Oh, okay, sorry. Now?
Yes, perfect. Good.
Okay. Good afternoon or good morning, everyone. My name is Song Rong. I'm a PhD student at Hasselt University. Today, I'm very glad to be here with my colleagues, Bridget, Rosella, Michelle, and Tim, and to give our presentation, Meet in the Middle meets multi-omics, a strategy to identify molecular signatures of environmental drivers of childhood BMI. We have two main objectives in this study. The first one is to integrate exposome data and multi-omics data, which are both high-dimensional and heterogeneous. The exposome includes both prenatal and postnatal exposures that are of various data modalities. And in the meanwhile, the multi-omics data are from 4 different assays with completely different dimensionalities. The second objective is to perform a causal inference addressing whether the multi-omics mediates the effect of exposures on the childhood BMI. We then show the study design in a flowchart. In the first step, we perform a multi-omics factor analysis, or in short, MOFA, for the multi-omics data and exposome data in parallel. We filtered the transcriptome and DNA methylome data based on each probe's relevance to the outcome BMI. And the DNA methylation, here we use the M values. The M values are denoised against the blood cell types variations. And then since the exposome data are even more heterogeneous, we simplify the model training by making them into 4 sub-exposomes based on whether a specific exposure is prenatal or postnatal, and whether it is a numeric or categorical variable. Through training the MOFA model, a group of low-dimensional latent factors are extracted to represent the major variation in the data. Then the next step is a downstream analysis. The BMI z-score is regressed on each of the multi-omics factors, and then we select the significantly associated factors as the reduced multi-omics. Then the reduced multi-omics in turn is used for the downstream analysis of the exposome factors. Then in a similar way, we select the reduced exposome. Then the 2 branches meet in the middle in a final mediation analysis. This is how we combine different methods as a workflow for our study. So let's first look at the MOFA on multi-omics. The 4 data matrices are the inputs for the model training, and each one is specified with Gaussian likelihood. The observations are not completely overlapping between the 4 matrices. Therefore, there are some missing values shown here in gray color. In a model training setting, we dropped the factors that explain less than 2% of the variance in the data, and therefore we obtain a 6-factor solution. This heat map shows the proportion of variance explained by each factor in each assay, and this bar plot shows the cumulative proportion of total variance explained in each assay. We can see that factor 1 explains the highest variation in gene expression, and factor 2 and 3 explains mostly the variation in the serum metabolome. And then also, if we regress the BMI z-score on each of the factors, we found only the factor 1 to factor 3 are significantly associated with BMI. Then in a similar way, we performed the MOFA on a exposome data. We specified Gaussian likelihood and the Bernoulli likelihood for numeric and the binary sub-exposomes respectively. And we extracted 3 factors, and factor 1 explains mostly the variation in the 2 numeric sub-exposomes. And these 2 numeric sub-exposomes have also more variance explained than the other 2 binary sub-exposomes. And in a similar way, the previous identified 3 multi-omics factors were regressed on each of these 3 exposome factors, and we found exposome factor 1 is significantly associated with omics factor 3, and exposome factor 2 is associated with omics factor 2. Then these 4 factors are used in a next step of mediation analysis. If we take a look at what the factors represent, we can plot the top 5 absolute factor loadings for each factor. And then if we now look at factor 2 of the multi-omics, it gives the urine trimethylamine oxide and some serum glycerophospholipid PC, and also the urine hippurate. And then if we look at factor 3, it gives also some glycerophospholipid, but this time it's lyso-PC, and also serum biogenic amine. And we also found CpG from the DNA methylation that is mapped to the RTKN gene. Then for the exposome factor, if we look at factor 1, it represents mostly the building densities and access to public transportations. And then factor 2 represents mostly the prenatal consumption of food like cereal, legume, and dairy products, and also the maternal smoking. And also there's one family affluence score. Then this figure gives, these 2 figures give a better overview of how to interpret the factors. And we can see in the left, it shows the multi-omics. We don't see a very strong driving variable for each of the factors, but we can see that relatively for factor 2, we can see like the urine hippurate, urine TMAO, and serum PCs. And for factor 3, you can see relative strong effect from also the serum lyso-PC and also biogenic amine. But we can see in comparison, in the right, the exposome, there are really strong driving variables. For example, like for factor 1, we can see in every sub-exposome, we can see the built environments contribute to factor 1. And for factor 2, there's almost only one strong driving force of maternal smoking. Then for the mediation analysis, we adopt structural equation modeling, in which we can incorporate multiple causal relationships in one and the same model. And considering the data comes from 6 different cohorts, we used a multi-level model and to account for the between cohort difference. However, our interest is mainly in the within level. So this is, if we zoom into the within level, this is what we have as the path model. And we have adjusted every model for these selected covariates. Then we have the model estimates. We can see that except for these 2 edges of the direct effect, the others are all significant. And the indirect effect and total effect are also significant. Then if we give a very brief biological interpretation of this result, we can say that like the effect of maternal smoking on the childhood BMI is mediated by some serum and urine metabolites. And also the effect of built environment on BMI is also mediated by some serum metabolites. So in conclusion, what we have done that we have used this novel workflow to combine multi-omics factorization with a meet in the middle strategy, and especially on to study the effect on childhood BMI. And we successfully integrated high-dimensional and heterogeneous data, and we have identified the main drivers of the variation within the data. And we found that the omics factors, especially representative of the metabolites, can mediate the effect of exposome factors on the childhood BMI. The strength of this method is that it's a simple and efficient way to for causal inference, because we only need to find the overlap between the biomarker of the exposure and the biomarker of the health outcome. And in all the models we used in this method, missing values are allowed. We don't need to do any list-wise deletion. And limitation is that this is only an explorative analysis. And in the future, maybe we can do some validation using subsamples, or we can use subsamples for different steps to make a more strong conclusion. Thank you for listening, and I'm happy to take questions.
Thank you very much for your presentation. Very, very interesting results with mediation. We have a question from Carl. So thank you for great presentation. Did you consider performing MOFA on the combined multi-omics exposome data?
We didn't try, because what we wanted to do is to see the mediation, and with this meet in the middle strategy, we want to see like separate effect within the exposome and the multi-omics. So we haven't tried to combine the multi-omics with the exposome. But it's worth trying if we can find, perhaps we can find more links between the exposome and multi-omics.
Yes, definitely. I think one of your latent factor, or what you call factor, is represent like socioeconomical class with the family affluence and the maternal smoking.
Yes,
so that's interesting. Carl said that it would be interesting to compare those approaches with the combining the omics and the exposome separately.
Yes, indeed.
Good. Well, if there is no more question, we'll move to the next speaker. So, our next speaker is Daniela Sugna from University of Turin. Daniela?
Yeah, hello. I'm sharing screen. Can you hear me? Yeah.
Yeah, we can see you and hear you.
Okay. Let me see if I can. Okay. Hello to everybody. Okay, in this presentation, I want to show you an application of this novel method for dealing with the high-dimensional mediators in the exposome context. I'm from University of Turin, and I want to thank Chiara Moccia for helping me to prepare this presentation. So, as probably all of you know, mediation analysis aims at estimating to what extent the effect of an exposure A on an outcome Y is explained by multiple mediator, by a set of mediator on this causal pathway. In the counterfactual framework, we can decompose the total effect of A on Y in the natural direct effect. It means the effect unexplained by the mediators, and the natural indirect effect. It means explained by these mediators here. But the extension of natural effect to the multiple mediator setting are complicated by the complex and unknown, very often unknown, confounding patterns among these multiple mediators. And most of the methods that have been proposed up to now can be applied only when the mediators are independent of each other, or can be order, causally order, actually. We can propose an order between the mediator. This paper proposed by Law recently in 2020 actually proposed to specify only a model for the outcome and no models for the mediators. And the mediators and outcome can be continuous or non-continuous, can causally affect one another, and can also share unmeasured common causes, actually. Then can be some unmeasured variable causing the mediator. And we don't have to take in account for the ordering of the mediator, and we can, however, decompose the total effect, indirect effect, and a joint indirect effect, and its turn, this joint indirect effect can be further decomposed in an indirect effect via each mediator, an indirect effect via the mutual dependence among mediators, and a so-called remainder indirect effect. When I talk about effect, I mean interventional effect. They are different from natural effect because the interventional effect consider population level intervention that set the value of the mediator to a random draw for its distribution that depend only on the exposure, not on the covariates, not on the other mediator, but only on the exposure. So what interventional direct effect expresses is the change in the outcome that we could observe if the distribution of the mediator were shifted from what it would be if all subjects were exposed to what it would be if the subject, for example, were unexposed or would otherwise be, actually. So actually, the interventional direct and indirect effect are relevant, for example, when we can intervene on a mediator, on the distribution of the mediator, and they can be identified also under weaker assumption than the natural effect, after I tell you in which way. So what we need to estimate is this potential outcome here. This is the potential outcome under A equals 0, when the mediator value are set to a random draw from the joint counterfactual distribution of the mediator under A equal 1. And this potential outcome under A equals 0, when the mediator values are set to a random draw from the marginal counterfactual distribution of the mediator under A equal 1. Then what we need to do is only to draw the mediator value from actually the joint distribution and marginal distribution, the counterfactual marginal and joint distribution under some exposure levels. As you can see here, both joint and marginal distribution for the counterfactual mediators are unconditional on the observed baseline covariates AL or confounder, as you prefer. And in this marginal distribution, the mediator are also independent from the other mediator. So we can estimate the total effect, which is as you can see, the only the difference in the total effect is in the exposure and also the mediator level for different exposure level. Here is the exposure is 1, here it's 0. However, it's the total effect of the exposure on the outcome. Can be decomposed in the joint direct effect, where I change only the exposure level, and the joint indirect effect, where we change only the counterfactual distribution of a mediator under the exposure level 1 and under the exposure level 0. And we can estimate also indirect effect with each mediator in this way. And as you can see here, I'm considering the marginal distribution of the mediator, and what it changes in this expression in these two terms is only the value for this mediator MS. It means that here I have to draw the mediator value from the marginal distribution of the mediator MS under the level exposure A = 1. Instead, here I draw the mediator value from the marginal distribution when the exposure is equal 0. The effect through this mediator MS is the combined causal effect along all paths from the exposure to the mediator. I mean that if we estimate the indirect effect through M3, for example, I capture this pathway here, A, M1, M2, M3, and Y, and A, M3, Y. But for example, I don't capture if there were a mediator here between M3 and Y, I cannot estimate. It doesn't capture the pathway, including subsequent mediator in the pathway to the outcome. So the assumptions are always the usual as for natural direct indirect effect. So the effect of the exposure on the outcome has to be not unconfounding, conditional on covariates. Then we don't have a measure confounding between the mediator and the outcome. And also we don't have a measure confounding between the exposure and the mediator. But we can still have measure confounding between the mediator and the outcome affected by the exposure. So the underlying assumption to identify and estimate this effect are weaker than the assumption to estimate natural direct indirect effect. What we need to do is to estimate the joint effect. We need to fit only the outcome model, conditional on the exposure, the mediator, and the covariates. Then we need to construct duplicated data for each individual in order to estimate, to randomly draw the counterfactual mediator value for each value of the exposure in order to obtain some random draws and then to estimate the potential outcome. We need to use a Monte Carlo approach in order to take in account for the variability in the counterfactual mediator value, and then we can calculate the average imputed potential outcome across all individuals to estimate the joint direct indirect effect. To estimate the indirect effect via the single mediator, we need to expand the duplicated data set and then to, for each row and for each column, to randomly draw the counterfactual mediator value unconditionally on the other mediator from each observed exposure group, and then to repeat the previous step, the Monte Carlo approach, and average the imputation potential outcome. When the exposure is not randomly assigned, we need also to sample the mediator value with the probability proportional to the inverse of the conditional probability of receiving the observed exposure given the confounder. Then we need to consider this inverse probability weighting. And the authors of the paper suggest also to use some penalized regression models for the outcome model as well as for mediator model in order to reduce the number of mediators, mainly when we have fewer observations than mediators and covariates, and to unify the two results of this penalized regression model, and then estimate the pathway of interest. In our example, we consider this population under study. Our exposure of interest is the socioeconomic position, low versus height. The outcome is the low birth weight, and we identify 91 potential mediators evaluated during pregnancy, air pollution, built environment, lifestyle, metals, and so on. And we identify these confounders here. I show you very briefly the results without going in detail in the interpretation.
Yes, please, 1 minute left because we are running out of time.
And we estimate the joint indirect effect, the joint direct effect. This is the effect on the additive scale, absolute scale, on log odds difference scale. And this mutual dependence is the effect which depend on the dependence between the mediator. Then how much the exposure affect the outcome by the mutual dependence between mediator. Actually, there is no evidence of an effect. However, the joint indirect effect suggests a positive effect here. Here I've included only the mediator for which we have found an association. For example, if we focus on traffic density and population density, traffic density, we can say that being low, having a low socioeconomic position has an harmful effect on the risk of low birth weight, increases the risk of low birth weight through traffic density. And it's quite important because it's an interventional effect. It say that if the distribution of traffic density were shifted from what it would be if our subject were low socioeconomic position to what it would be otherwise in high socioeconomic position, then we could decrease the risk of low birth weight. Actually, then we can intervene on the single mediator in order to decrease the indirect effect. Here, only the plot of the other mediator we have considered, and we didn't find evidence. I want to underline that there are many areas of future work of these methods. We need to establish theoretical property to ensure valid inference of the Monte Carlo based estimator following mediator selection. And we can also compare different methods to select the mediator in order to see what happens in this method. And we need to assess the robustness to if we misspecify the outcome model. And also to include missing data. Actually, we have performed the analysis on complete data. Okay, thank you very much. I guess from.
Thank you, Daniela, for this presentation. I like the diversity today of this session, actually, of the different methods proposed. It's very good. Is there any question from the audience? There's only one presentation left after this one. No? Daniela, I have one. There is a question from the chat. Is it possible to account for potential interactions between the you want to read it yourself? Maybe can you see the chat?
Yeah, I can see the chat. We cannot. We don't need of linearity assumption. Absolutely, I included both binary and continuous variable. Then we don't need also the outcome can be continuous or not continuous as well as the exposure. Actually, only the method is different. Then it's really flexible, and we can include absolutely the interaction between exposure and mediators in the model for the outcome. We don't need to model the mediators actually. Then only if we want to apply the selection methods to select a set of mediators, then we can propose, we can include the interaction between the mediators. But otherwise, we can include the interaction between the mediators only in the model for the outcome, because it's the only one we need to explicit correctly. Actually, yeah.
Okay. Thank you, Daniela. We will move to the next speaker. The next speaker is from Icahn School of Medicine at Mount Sinai in New York. Miao Yu, please. Can you share your slides?
Could you see my slides now?
Yes.
Okay. Hi, everyone. Good morning or good afternoon or good night. So I'm Miao Yu from Icahn School of Medicine at Mount Sinai. Today, I want to share an idea about molecular gatekeepers bridge the exposome and health. So when we talk about how the exposure influence a certain health condition, is actually we want to know why. That means sometimes we can see the association between the exposures to the disease, but finally we want to know why this happens and which give these effects. So here I choose metabolites because of course we can change to other omics data, but are all the metabolites related to this process? So I want to propose a theory called gatekeepers. Imagine all the exposures as different kinds of virus, and finally is some disease like COVID. So what? So a virus, if we want to cause to a disease, it must attack to ACE2, these receptors. So I want to know, is there some metabolites play as the same roles as ACE2? So these metabolites must associate with exposure, also associate with other metabolites that not directly associate with exposures. So I call it gatekeepers molecules, but this molecule could be metabolized or gene or protein or some others. Let's think it in details to figure out what's the pattern of them. Imagine all the metabolites, there are actually only 4 types of them. One type is metabolite that has no association with any other metabolites or the exposures. This is like some isolate. And some metabolites, they only associate with exposures, but they will not influence other metabolites. But still, there are some metabolites that not only associate with external exposures, but also with some other metabolite clusters, which seems like a continent. And some metabolites, they are both associated with the exposure, and they also associate with each others. So what is gatekeepers? The gatekeepers is actually these triangle ones. This one, they always has rules between one exposures and another metabolites, whether these metabolites are directly associated with exposure or not. So here the task for gatekeeper discovery is quite simple and clear. We just want to screen all the structures like this. So we can then check whether those gatekeepers are more sensitive to a certain disease or not. So this is more like a hypothesis generation process. So to discover those gatekeepers, there are clearly 3 steps. The first step is we need to check the correlation among all the metabolites, whether by a network analysis or some other method. Here we just use a Pearson correlation, paired Pearson correlations, and then we connect them if they has a high correlation coefficient larger than 0.9. And then we will check all the association between metabolites and the exposures to screen the structures. And after we select those so-called gatekeepers, the next step is we will make the association between those gatekeepers and health outcome to see whether they are sensitive to certain health outcome or whether they have some general rules to respond to multiple different exposures. So here is what I use the sample. The sample is from the shared data, and we only use the serum metabolome and 177 metabolites for it. And for the exposures, we use 117 continuous variables. And for the health outcome, we have a very simple selection, only Ashma or not. So here, first we check the correlations. Since this gatekeeper theory is actually developed for untargeted metabolites, metabolomics, for this target, the network is quite simple. We can say that it's actually only one large network. All the others are just paired networks. And most of the metabolites actually has no correlation with other metabolites, which means they are more like some island instead of a continent in the metabolites level. And then we will go to the next step to check the metabolites and their association with the exposures. Here we can we checked and found 155 peaks among all the metabolites. They are only connected with exposures, but there are still 22 metabolites connected with both other metabolites and exposures. This is what we want to know. But in this network map, I only displayed 12 of them. This 12 is more different. They are not only connected with one exposure, they connect to multiple, at least 2 different exposures, which means they has a general response to different exposures, which is more like the rules of gatekeepers. So here we can see some interesting structures, like this one is a gatekeeper. It stops the influence from these 2 exposures to these metabolites, and also some others. And this metabolites is protected by different gatekeepers. So the next here is a summary of how these gatekeepers associate with different exposures. Here we can see that some metabolites are actually associated with quite a lot of different exposures, which means they just play some general roles to give a response to outside influence. And also, if we want to check the exposure levels, actually there are some exposures with association larger than 6 gatekeepers. 3 of them are PM10 rated exposures, and 2 of them are metals, and one is PCB. And another, I think previous presenter also mentioned, is NDVI listed index and some other access. So after checking this, we just screen out these exposures. The next step is check whether those gatekeepers could be sensitive to some health condition. So here we check actually it's just a t-test between gatekeepers and health outcome. We can find that 3 of them actually can distinguish whether they have the asthma or not. Which means this may be some general rules for those 3. And I want to go to the conclusion part and treat the gatekeepers as some battlefront metabolites between exposures and other metabolites. If we can screen them, we will always find that some gatekeepers are sensitive to multiple environmental exposures, which implies that for exposome study, we may have a much smaller subset of metabolites level compounds that can respond to multiple different exposures. And also we can see that those gatekeepers may reveal some details about the relationship between the exposome and the health outcome. And it can also help to generate some hypotheses for future study. For example, we know that in metabolite level, there is a symbiotic metabolite pathway. But I think by using the gatekeeper framework, data analysis framework, we can actually find more different kinds of this symbiotic pathway, which may not be revealed before. So the gatekeepers here is a molecular level concept. It can be metabolized, but it can also be gene, protein, or some others. In the shared R markdown scripts, I also analyzed the gene. Actually, we can find almost 90 gatekeeper genes that they play like a protected roles between exposures and other genes. So if we have those gatekeepers at different levels, maybe an idea to connect the different omics is just to connect those gatekeepers because they may have a different rules in to respond to the outside exposures. So so this study is actually done in Dr. Patrick's research group. This is our group's pictures, and this is Dr. Patrick. And we also get help from other collaborators, and we will release a manuscript maybe next month for molecular gatekeeper discovery for multiple exposure groups in adolescent girls. And we get funding from the NIHS for this kind of study. So thank you. So this is all of the concept of gatekeepers. And if you have any questions, feel free to contact me either via email or through the GitHub account. So yeah.
Thank you, Mario, for this presentation. You actually have some question in the chat. I will read to you. There is one from Xiatao, who says great work, and he's asking if you use all the thousands individual to calculate your serum metabolite correlation.
Yes. Yes, we use all the data that's shared in the I think by this project.
Yeah, it's around 1,200. Yeah. Okay. And Carl is asking, saying it's a really cool concept, although it's based on univariate analysis. So do you think that there could be other underlying phenomena behind gatekeeping, such as interaction between different omics variables?
Yeah, for sure, there should be. I mean, this is just a very simple idea. Actually, it can be coupled to different, I think it can be coupled to different workflow as long as we can find some phenomena to support there to have this phenomena. I think there will be some interaction between different omics variables, but how to connect them, I didn't discuss here, but maybe we can discuss for, we can use different models to select the association between exposome and the metabolite. For example, we cannot, here I just use a linear regression to select the associations. We can use random forest or other method to coupled with, as you said, non-linear effects or some other different kinds of situation. We also need to consider covariance, but here I just use a simple, as you said, a simple comparison, didn't consider the covariance.
Yeah, yeah, I like the terminology you're using here. It's quite refreshing on gatekeepers and battlefront. Very good, very creative. Is there any other question specifically for Mario? Otherwise, we can move for the general discussion. I think I would like to start the general discussion, like actually maybe, yeah, moving on from your presentation. I'm thinking in particularly metabolites, how some of the results that come out, like for example, some of the metabolites, they're close to the phenotype, for example, to BMI, and they actually they may be a consequence of obesity, and they will have maybe correlation, association with like more general covariates that are associated with high BMI, like maternal education or diets, but diet is not influencing directly the metabolite we're measuring. It's just a metabolite can be seen as fine tuning of the phenotype itself. But there are also the omics markers, they can be closer to the exposures. For example, in the serum metabolite, we measure metabolites that are more related to fish intake, like omega-3s. We directly measure omega-3s, and you will see that they are associated maybe to an exposure, but it's all related to families that have a lot of high fish intake. And there's no causality there. So I don't have any answer. I don't think there is one answer, but I'm just wondering, how do you, in mediation analysis, how do you take that into account, that the omics layers, they might be representing more a consequence of the phenotype or a source of the exposure, but they're not actually in the middle of the exposure and the phenotype. They are more representative of one or the other, just fine-tuning the exposure or fine-tuning the measurement of the outcome. I don't know if...
There are so many questions in this that you're asking. I mean, if we start by looking at the serum metabolome that is measured, then that is an incredibly biased selection to start with. This represents one panel with metabolites that are the usual suspects, basically. That's why they are in this panel. So that's a problem in itself. It's not an unbiased picture of the metabolome. So if that picture was unbiased, we would expect there to be a sort of wild distribution between stuff related to exposure, stuff related to outcome, and anything in between. But here we know that a lot of these will be related to phenotypic outcome, because that's what they are selected for. So I would guess, and with the exception obviously of omega-3 fatty acids and a few other candidates, I would expect most to be more on the outcome side of associations. But there's another fundamental or philosophical question in this. Does mediation analysis really address causality? Can we really use this type of data to infer causality by using fancy statistical tools? And I'm rather skeptical to the potential of using a mediation analysis for these purposes. They can give you an indication, but just because that there seems to be a mediation effect does not mean that it's actually mediated either. So I mean, we can generate. I think we should be always very, very careful in how to interpret data from these kinds of studies and exploratory research, and really always keep that open. We need to confirm this in other studies. So it's not an answer, sorry.
No, but I expected something like that, just more brainstorming on this. Yeah, because I think some of the omics can be also used as better adjustment factor for the association between the exposure and the health outcome. Something we haven't measured through questionnaires or, yeah. Xavier, yes?
Yes, no, a bit on what you were saying. In the paper I shared in the chat, we look at actually different causal structures and then testing different methods that are used to see if there is a middle layer that can help in identifying the effect of exposure on a health outcome. And we found that, well, if the truth causal structure is one of reverse causation, where basically what you are considering to be a mediator is just a consequence of the health outcome, there are some methods that work better to avoid that. But of course, there are multiple potential causal structures, and you never know beforehand which one is the true one. So it's difficult to decide what to do. But yeah, in that paper we provide a bit of guidance on things that could be done.
Doesn't that really pinpoint the issue that when you know the true structure, it's easy to find the method that describes the true structure, but we don't know the true structure for most of these things, really, and especially if you go to the full untargeted metabolome, where they are not. There are other biases in untargeted metabolomics, of course, but that you can only measure what the analytical instrument can measure. So therefore, we limit to certain classes, or some classes are overrepresented, but it's not based on a literature reporting bias to the same extent. So then the ground truth is not known to the same extent.
And when you get to,
sorry. Yeah, sorry, no. Go ahead, Leah.
No, and it's even more complex when we're going to exposome data, because if you just look at, I don't know, heavy metal, fish, and IQ, okay, it's easy. You can have your DAG, you can have your hypothesis, but now with exposome, with from many domains, it's much harder to do, like to use your prior knowledge for this causal structure.
Yeah. Can I only say one thing? I think that a benefit of the method proposed by law is that at least you don't have to model you don't have to draw the causal structure between mediators, at least, because actually really when you have many mediators, if they are many mediators, then you have to consider also that obviously, but at least you don't have to draw the causal structure between the mediators, no, because obviously as the number of mediators increases, so it's really difficult, and probably yeah, obviously anybody has an a priori idea different from the other, then probably everybody would disagree about your DAG. Then yeah, I think this is a benefit, but yeah, it's really difficult. Yeah, I totally agree.
But in the method used by Carl, I felt somehow when you start to add all the omics together, maybe they adjust somehow for each other limitation. If you have the metabolome, for example, that represents more the BMI of the child, and methylation is closer to the immune system, then you start to see more patterns.
Yeah, and I think that addressing multi-omics or different limitations, if you can integrate data at a very, very early stage, as close to the raw data as possible, you actually have better chances of finding interesting things. Obviously, you're also contributing to the underdetermination of your mathematical system. So there are other complications when you start to do that. It's always, you know, trade-offs. If you gain something from doing it one way, you lose something else. And in our case, we find that it's mostly costly in terms of computation. So we still don't find a lot of evidence of overfitting, but you really need to think carefully about what you're doing. And even with complex data analytical architectures, the devil is in the details. Each and every module that I showed during my presentation has a bucket load of limitations, that we do machine learning to select variables because they interact. But then we adjust for covariates using linear modeling. So then we disregard what we believe to be truly important, which is the interaction between variables. So we're working on trying to develop the next generation models where we can put this into one unified, into better models. But really, I think we should try to work on combining different models way more than we do. Not working on grand unifying theories, one method to rule everything. So sorry, I went off on a tangent because you were saying something else, but I came into my pet peeve. Don't do one model, do many models and combine them in intelligent ways.
I like this. Are there comments from the rest of the audience, from the other speakers? No, otherwise we can close here. I will see you again tomorrow. Thank you very much for the presentations today. I'm so impressed by the quality and the clarity, even if you presented like quite complex methodology sometimes. Yeah, very interesting results, and maybe we can take time tomorrow to discuss a bit more. Yeah, what's our future steps and future direction. Thank you, everyone. Have a good evening, a good rest of your day. And I'll see you again tomorrow at, sorry, I forgot. Yeah, at 2:30. And we don't have a keynote tomorrow. We start straight with the last topic, and we'll have concluding remarks from Xavier. Bye, everyone. Thank you.